यार मित्र बोलूया अरे ना यार බලන්න යාර විතර පේනවා පොඩ්ඩක් මෙහාට වෙන්න
Good morning, all. Warmly welcome you all to the second day of 9th Annual Scientific Session and International Conference contributed by Sri Lanka Association for Laboratory Animal Science. Moving to today's agenda, first of all, I would love to introduce our honorable judge panel, who is responsible for the whole day program schedule, including 3 sessions to be continued. Dr. Anura Jayasuriya, Senior Lecturer, Department of Basic Veterinary Science, Faculty of Medicine and Animal Science, University of Peradeniya. Professor Nayana Vijayawardena, Head of Department of Clinical Science, Faculty of Veterinary Medicine and Animal Science, University of Peradeniya. Professor Charita L. Gunasekara, Head of the Department of Biochemistry, Faculty of Medicine, Kothalawala Defense University. Please give your applause to welcome them. And I cordially invite Dr. Anura Jayatilaka to come and sit the head table. As our first program event, we are ready to start free paper session part number 1. I would love to invite the session chairs, Dr. Mayura Thammitiyagodage from Medical Research Institute, and Dr. Ureshini Karunaratne on behalf of University of Jayawardena to host and proceed the session. We have to wait a little bit until Professor Nayana Vijayawardena and Professor Charita L. Gunasekara, who is going to join today's sessions in virtual online way.
Good morning.
Good morning to everybody. Any scientific session, technical session is the heart of the, scientific component of research. Actually, this is related to laboratory animal science, and there are a lot of young scientists who are engaged in laboratory animal science in the country, as well as alternatives. This is an excellent opportunity to, share their experiences while doing the experiment. That is the practical exposure. There are, in the first technical session, there are 3 papers lined up, and Dr. Ureshini will invite the presenters to deliver their presentation.
Very good morning to you all. Welcome to the free paper session 1 of 9th Annual Scientific Session of Sri Lanka Association for Laboratory Animal Science. So today we have 3 papers for the first session. So so all the presenters, there are 10 minutes were allocated for each presentation. There is a one bell ring once you pass 8 minutes, and is a two bell ring once you pass the 10 minutes. Okay, so you should have to be in alert to finish your presentation on time. So I would like to invite our first presentation. So our first presentation is on effect of NF-kappa B inhibitor SN52 on Frankel-induced osteoclastogenesis. The authors are Prasadi MGTM, Prematilaka EGCN, and Alas CNRA. Today, the presenting author is Prasadini MGTM. So I would like to invite Prasadini to proceed with it.
Good morning, everyone. My presentation is about effect of NF-kappa B inhibitor SN52 on rank ligand-induced osteoclastogenesis. So, bone is a dynamic organ. In adults, maintenance of functional and structural integrity of bone is regulated by balanced action between osteoblast and osteoclast. The main function of osteoclast is bone resorption. Human nephrotic factor and human nephrotic factor receptor superfamilies together regulate osteoclast differentiation and activation. Activation of nuclear factor kappa B pathway by rank ligand is important in osteoclastogenesis. NF-kappa B transcription factor family, which is consisted of 5 structurally related members, including NF-kappa B1, NF-kappa B2, REL-A, REL-B, and C-REL. Nuclear factor kappa B act by 2 main pathways known as canonical and non-canonical pathway. In canonical pathway,
We can see the other side. Forget about that and proceed.
In canonical pathway, external stimuli like rank ligand activate I-kappa B IKK complex. This IKK complex then activate P52 and REL-B dimer by inhibiting NF-kappa B molecule. Then translocation of P50 and REL-B complex cause osteoclastogenesis. In non-canonical pathway, IKK alpha dimer also important in activation with NIC, NF-kappa B inducing kinase, and cause activation of P100, and then P100 partially degraded into P52 active product, and P52 REL-B nuclear translocation leading to osteoclastogenesis. Here, in literature says rank ligand inhibition is a valid approach for inhibition of osteoclastogenesis, but there are adverse effects of long-term use of rank ligand inhibitors in immune system. So we need to inhibit the pathway where the immune cells are not caused any adverse effects. So in this current study, we use an SN52, which is a specific REL-B inhibitor, which is a synthetic cell-permeable peptide which selectively blocks the non-canonical pathway through inhibiting nuclear translocation of REL-B. as well, that cause accumulation of P100 because in this level, SN52 axon LB and P52 dimer, this cause accumulation of P100, and P100 goes to interact with P50 and REL A complex in canonical pathway, also cause inhibition of osteoclastogenesis. The main objective is current study is to investigate the inhibitory effect of NF-kappa B SN52 on osteoclastogenesis. The secondary objective is to assess the viability of osteoclasts treated with SN52. In this diagram, bone marrow stem cell isolation is brief. In this diagram, bone marrow stem cells were isolated from tibia of male biopsy mouse, and then cells were plated in 25 square centimeter corning flask in culture medium supplemented with macrophage colony-stimulating factor, and then incubated at 37 degrees of Celsius with 5% carbon dioxide for 48 hours. After 48 hours, osteoclast precursors are isolated and reseeded and placed in 48-well plate and 96-well plate for two different experiments. In 48-well plate, the day of osteoclast precursors are harvested as known as day 0. In day 0, in 48-well plate, at a rate of 1 into 10 to the power 5 cells per well, we cultured it media supplemented with macrophage colony-stimulating factor, Rank ligand, and 3 different concentrations of SN52. And here we use a control sample, and all the samples were triplicated in this study. After supplementation, we incubated the cell culture plate for 72 hours. After 72 hours, we changed the spent culture media, then again supplemented with MCSF, Rankel, and 3 different concentrations of SN52, same as the day 0, and then again incubated for another 48 hours at 37 degrees of Celsius, 5% carbon dioxide incubator. At day 5, we identified osteoclast by staining for TRAP. In cell viability assay, in day 0, we reseeded the cells in 96-well plate at a rate of 1 into 10 to the power 4 cells per well in alpha MEM medium with 10% FBS, and we incubated the plate for 24 hours. Then after 24 hours, we removed the spent culture media and wells were treated according to this table. And again, we incubated for another 24 hours at 37 degrees of Celsius with 5% CO2. And after removing the spent culture media, we added MPT reagent and incubate for 4 hours in dark. During this incubation, we observe under microscope for development of any purple color crystals. And after 4-hour incubation, again we added DMSO, dimethyl sulfoxide, and incubated another 3 hours until development of consistent purple color. After that, we took spectrophotometric reading and calculated for percentage of viable cell count, using a calculation. This is the results of in vitro study of osteoclastogenesis. Here we can see the TRAP positive multinucleated cells, which are more than 3 nuclei. Here I indicated with arrowhead. With the increment of SN52 concentration, the number of mature osteoclasts is reduced when compared with control. I think you can see those in, and in table 2, we can see the number of mature osteoclasts per unit area is reduced with the increment of SN52 concentration. In table 3, we can see number of immature osteoclasts per unit area is increased with the increment of SN52 concentration. This is the results of MTT assay. Here we can see there's no significant, statistically significant reduction of viable cell percentage. In my discussion, referring to previous studies, SN52 plays an important role in downregulating the NF-kappa-B alternative pathway of osteoclastogenesis. The results of current study reveal number of mature osteoclasts reduced significantly with the increment of the concentration of the inhibitor compared to the control. And in MTT assay, there was no statistically significant difference in cell viability assay. So the conclusion, there was a significant reduction of osteoclastogenesis with SN52, which is a specific FeLB blocker, and there was no SN52 toxicity observed in osteoclast. Thank you. These are my key references. Thank you.
Thank you, Prasadini, for your interesting presentation. And now the presentation open for the questions. So while we are asking the question, you can place the laptop here.
I have one question.
Yes,
In
ma'am.
your presentation, you said that you use male BALB/c mice to collect the bone marrow.
Yes, madam.
Is there any specific reason to collect male bone marrow?
Yes, madam. In female animals with the age, the estrogen concentration will different. So I want to maintain the constant level of hormone. So I took the male animals.
At what age you selected those male animals?
5 weeks old.
5 weeks old, female and male are not, I mean, there is no age difference at that age. I don't think even there's a use male mice, BALB/c.
Yes,
I don't think at that age there will be a significant difference. But anyway,
yes.
my other question is, what is the statistical package that you use to analyze your cell culture data?
SPC.
Statistical technique.
ANOVA method using SPSS software. madam.
ANOVA, yes.
Yes.
One way, what is the specific statistical, I mean, t-test, or what is the
Student details.
student? Okay. Thank you.
You used NF-kappa-B inhibitor, no?
Yes, madam.
Is it a
Yes, sir.
synthetic one?
Yes, sir.
So what is the application of using this one? And can it be used for treating something, pathological conditions?
Still we didn't the molecular pathways. So using
But
Western
your
blot
aim is to inhibit the osteoclast, no?
Yes, sir.
So bone resorption, and is there a relationship to osteoporosis application?
We are looking for that matter, sir,
Mhm,
in my animal model study.
mhm. Because in your presentation, it's better if you just, describe the audience about, the general information, the application and all these things in introduction.
Yes.
So anyway, it's good presentation and you've done very well. Okay.
Can I ask one question?
Any question from audience?
Can I ask one question, please?
Yes, madam.
Yeah. So actually, Professor Andrew also mentioned the same thing. So I would like that if you had described the significance of your study briefly, right? So can I ask you what is the significance of your study? Like, if someone asks that, how do you explain that?
I'm using this in vitro study results for do the animal model in vivo test, madam.
How representative would this animal model be with the human osteoclasts? Like in a human situation.
The thing is, madam, we are not observing the pathway in molecular level.
Mhm.
In literature, this is a proven NF-kappa-B inhibitor, RELB inhibitor, specific inhibitor as a SN52, madam. That's why we use this in this pathway. Without any Western blotting or other testing, we can't say this is can be used for humans, madam.
Okay. Okay, yeah. Thank you very much.
Are there any questions from audience?
The last question. A nice work of your work in time to come. Maybe your one is still at the preliminary level. As Dr. Jayasuriya mentioned, can you at least hypothesize what would be the clinical implication of your future, of this work in future?
We are using this as a possible drug target for osteoporosis in ovary-ectomized mice model, madam. The therapeutic implication in other animals and humans.
Okay,
It should be.
that's what I was. Okay, that's what I was looking for. Thank you.
Thank you.
Thank you, Prasadini. So we'll move to our next presentation. Our next presentation is on preliminary study on alteration of hepatic macrophages by crude leaf suspension of Osbekia ofrandra in Bovitia in thioacetamide-induced rat liver cirrhosis. The authors of the papers are Bogavatta B.R.S., Kodituakku S.P., Vijay Gunawardana MPB, and Vijay Surendra KK. Today the presenting author is Vijay Surendra KK. We would like to invite Mr. Vijay Surendra KK to present his presentation.
Okay, good morning, ladies and gentlemen. And our presentation is on preliminary study on alteration of hepatic macrophages by crude leaf suspension of Osbekia ofrandra, which is also known as Heen bovitia, in thioacetamide-induced rat liver cirrhosis. So to give an introduction to our presentation, actually mammalian liver, so that is a vital organ, which is involved in almost all metabolic pathways and one of the important organs inside the body. At the same time, the liver cirrhosis has become a big health burden globally as well as locally. And liver cirrhosis is mainly caused by repeated fibrosis after the liver injury. And normally that would be the endpoint of all chronic liver diseases. So when you look at the pathogenesis of liver diseases, and especially liver cirrhosis, there are two main types of cells are involved. One is the macrophages and the other one is myofibroblasts. With the, the synergy of these two cells, the fibrosis or the cirrhosis will be occurred. So at the same time, there is one antibody or a protein which is used to identify macrophages, which is known as ionized calcium binding adapter 1, which is widely used to identify the macrophages in livers. So the main objective is to check. So over here, what we wanted to do is to identify the effect of Osbeckia octandra in resolution of fibrosis. Therefore, we used aqueous extracts of Osbeckia, which has been widely used in traditional medicine in order to resolve hepatitis as well as certain liver conditions. And recently, of course, we found that feeding aqueous extracts of Osbeckia octandra, so that depletes thioacetamide-induced liver cirrhosis. So over here, we induced cirrhosis through thioacetamide injection, and that is very similar to human micronodular cirrhosis. However, we found that there is a resumption of cirrhosis, but the exact mechanism is not known. So therefore, in order to identify the macrophage kinetics in thioacetamide-induced rat liver cirrhosis, we performed this study. So in order to perform this study, we had to prepare crude leaf suspensions of Osbeckia octandra. For that purpose, we used 5 grams of leaves of Osbeckia octandra. Then we washed them with distilled water and air dried. Then we performed freeze drying, and then they were ground and sieved at 0.5 millimeter. And in order to feed to rats, what we did was we dissolved 1 gram of this ground and sieved freeze dried powder with 20 milliliters of distilled water. Right. So this is how the animal experimentation was conducted. We used male Vista rats, 6 weeks old, and we divided them into 4 groups, each group having 4 animals. So group 1, normal saline was given, normal saline was injected, and an equivalent amount of distilled water was orally gavaged. Then for group 2, so this is the TA only group or the cirrhotic group. So they were injected with thioacetamide, 100 milligram per kilogram body weight twice a week intraperitoneally. An equivalent amount of distilled water was fed orally. Then for group 3, the test group, so both TA and crude leaf suspension was given. So that purpose again, TA 100 milligram per kilogram body weight, and the crude leaf suspension was given at a dose of 500 milligram per kilogram body weight orally, and again twice a week. Then finally, in order to check whether there is any toxic effects or whether there is any solitary effects of crude leaf suspension on macrophages, we gave these rats injected with normal saline and crude leaf suspension at the normal dose. So then the liver samples were collected at week 5, 10, and 15, and the liver sections were fixed in 10% neutral buffered formalin and Samponis solution. Right, so this shows the results of our study. So this shows the gross pathology. So the control livers shown over here, and the rats treated with crude leaf suspension only, they were showing the normal hepatic architecture with smooth surfaces. However, the rats that are treated only with thioacetamide, they developed the cirrhosis, which is very similar to human micronodular cirrhosis. And numerous numbers of variable sized nodules were seen over here. However, interestingly, the rats that were induced cirrhosis with thioacetamide and fed with crude leaf suspensions were having cirrhosis, but to a lesser extent. So that is evident over here. There are very few numbers of nodules in comparison to the cirrhotic kidneys. So this is more evident in the histopathology, which is done with hematoxylin and eosine. So these control livers and the rats treated with crude leaf suspension only, they were showing a normal hepatic architecture with intact central veins. However, the rats treated with thioacetamide only, there were numerous fibrous bridge formations and large areas of collagenous deposition could be seen. However, the rats that are treated with thioacetamide and crude leaf suspensions, they were having less amount of fibrous bridge formation in comparison to the cirrhotic rats. So in order to check the fibrous bridge formation, we stained it with azan malari, and we observed in the rats that are treated only with thioacetamide, there is vast areas of collagen depositions, which are shown by blue color over here. And there is comparatively reduction in the collagen deposition in rats induced with cirrhosis and treated with crude leaf suspension. So the control livers and the rats that are treated with crude leaf suspension only, the fibrosis or the collagen deposition were just restricted to around the central vein and the periportal areas. Right, so in order to check the macrophage kinetics or the behavior of macrophages, we performed immunohistochemistry with IBA1. So in control livers, there were very few macrophages, mainly the resident type of macrophages. They were seen mainly around the central lobular region. But of course, over here, you can see the cirrhotic livers. There are many macrophages. There are some large number of macrophages could be seen, and some of them are lying on the developing fibrous bridges, as well as they were found on the liver nodules as well. So in rats treated with TAF and crude leaf suspension, there's a reduction of number of macrophages, and they were mainly seen around the fibrous bridge formation. Then in the rats with crude leaf suspension treated, so there were again reduced number of macrophages. So this shows the kinetics in a graphical manner. So in week 5, week 10, and week 15, in comparison to control and crude leaf suspension groups, there was a significant increase of IBA1 macrophages in thioacetamide induced and thioacetamide and crude leaf suspension treated rats. However, there was a marked reduction in the number of macrophages in thioacetamide induced, cirrhosis induced, and crude leaf suspension treated mice, rats in all three groups. So with these findings, we came into a discussion that Osbeckia octandra crude leaf suspension reduces thioacetamide induced liver fibrosis or cirrhosis, probably through altering the number of macrophage infiltration. So the importance of this would be, this would shed some light inventing novel therapeutics to the control of cirrhosis, because there is no any promising treatment for cirrhosis at the moment, except liver transplantation. So this study is actually supported by the National Research Council and the University of Peradeniya research grants. So these are my references, and thank you so much.
Can I ask a question?
Online.
Yes, please go ahead.
Excellent work, Dr. Vijayasundara. I just wanted to know whether you had received the ethical clearance for this. I'm sure you
Yes, we have received ethical clearance from the Faculty of Veterinary Medicine and Animal Science.
Right. My second question would be, is this heme bovitia currently being used as a Ayurvedic medicine?
Yes, that has been used as what we call as a kanda. So that kanda is made and used for hepatitis mainly, not for cirrhosis, but for hepatitis it is widely used. Okay,
probably your findings would give scientific insight into that, right?
Yes.
Yes, thank you.
Thank you, sir.
I also have one question, Dr. Vijayasundara.
Yes, madam.
Actually, this of heme bovitia or Osbeckia octandra had been widely studied in Sri Lanka. In previous studies, they have used CCL4 to induce hepatocarcinogenesis. Now here you have used thioacetamide.
Acetamide.
Yes. Is there an advantage over CCL4?
Yes, because there is when you look at the action, mode of action of thioacetamide, It is mainly of interfering with glutathione S transferase, which is mainly toxic induced one. I don't think there is much difference between carbon tetrachloride and thioacetamide, but it is said, I mean, recent publication says that thioacetamide induced cirrhosis is much similar to human micronedular cirrhosis.
Agent is a painful procedure.
Yes.
So for animal, they develop ascites and all that. So any painful signs while doing this?
Of course,
experiment?
no.
Can I ask a question?
Yes, please go ahead.
Yes, it was a very clear presentation. I have one question. Now you have used thioacetamide to induce cirrhosis. And I'm just wondering whether this treatment you used was also like concurrently with the induction. Like, did you use thioacetamide at the same time as the test drug?
Yes,
Or
yes.
you induce and later treated with the test drug and see whether the cirrhosis can be reversed?
Of course, no. We use this as a prophylactic treatment. So therefore, we used it concurrently, actually. And of course, another study we are doing by giving thioacetamide induced cirrhosis. And of course, we stop and we continue with the treatment. Probably we would be able to present it next year.
Okay, that would be very interesting to see, isn't it? Thank you very much.
Any idea about phytochemicals which are responsible for the action that you showed?
Of course, no, we haven't studied on that one, sir. But there are certain publications
Literature?
on that one.
In the literature, have you found anything?
Yes,
Yeah.
there are many on literature,
What
actually.
kind of?
Of course, sorry, I could not remember, sir, that one. Yeah, mainly antioxidants, actually. Yeah.
Thank you, Dr. Vijay Surendra.
Thank you
And
so much.
I also have a question. In our procedure, I saw that you have direct or did you obtain the suspension or what extraction from the involved?
No, actually, the leaves were taken and they were freeze dried, then made a powder. And the powder was mixed with distilled water.
In the Ayurveda activities, they use as a candle.
Kanda, yes, boiled one.
So is there? So I think there may be some effect because when we are preparing the candle, we heat those the leaves. So if you are using the leaves directly and freeze dry the sample, it may affect on the compounds that is present in the leaves. So there should be an effect.
Yes,
think,
yes.
Because the phytochemicals can be changed.
Change, yes, that's true. Because if in order to answer your question, actually, we did this experiment and it is already published. In that one, of course, we used 4 different extracts: boiled leaf extract, crude leaf extract, and methanol extract, and the other one, I guess, ETH extract. And we found the crude leaf extract is giving the best results. So that's why for this one we used crude leaf extract.
Okay, thank you very
Thank
much,
you so much.
Dr. Vijay Surendra. And now we'll move to the next presentation. The next one is on Artemia salinea, a replacement to animal model to evaluate the acute toxicity of Lephorisia hetmani sponge extract. So it will be the authors of the paper are Kurupwarachchi KASU and Gunathilaka KVK. So our presenting author is Kurupwarachchi KASU. Over to you.
Good morning, everyone. I'm Sashini Kurupwarachchi from Department of Zoology, University of Sri Jagadhanapura. So I'm here to present the abstract under the title of Artemia salina, a replacement to animal model to evaluate acute toxicity of Lufariella herdmani sponge extracts. So this is the content of my presentation. So sponges are considered to be the richest source of marine bioactive compounds. So besides from their bioactive properties, sponge extracts are also associated with toxic compounds due to their long evolutionary history. So these toxic compounds are potential drugs for a wide range of diseases, including cancer, but unfortunately, only a handful have been currently developed into commercialized viable drugs. So one of the major reasons for this may be due to the ethical issues of using higher animal models in early phases of toxicity screening. So there are alternative biological methods such as in vitro cell cultures, invertebrate models such as Artemia salina, zebrafish, and many more. So out of them, the Artemia salina is a well-known, simple and cost-effective model, which is widely used to analyze toxicity of natural extracts, which does not require any special training or resources. From this current study, this current study is an preliminary attempt to investigate the acute in vivo toxicity of the crude extract and the hexane extract of previously understudied sponge species Lufariella herdmani along with its zoochemicals and their percentage yield. So under methodology, this sponge sample was collected from Unawatuna, Sri Lanka, and the type specimen is deposited currently in the Museum of the Department of Zoology. And this sponge species was identified as Lufariella herdmani by using both morphology and the skeletal analysis, and by following the Hooper and Van Soet and the updated nomenclature in Van Soet et al. 2021. To obtain the sponge crude extract and the hexane extract, approximately 200 grams of sponge materials were diced and incubated in respective solvents as shown in the given slide, and for 72 hours, followed by rotaevaporation at 40 Celsius. Then the resulted extracts were evaluated, was used to evaluate the percentage yield and also to qualitatively evaluate zoochemicals by following standard methods with minor modifications. So under Artemia salina lethality assay, first the Artemia cysts were hatched under continuous aeration and illumination in a hatching chamber. Then the sponge, then the extracts were dissolved in 5% ethanol to get the ethanolic extracts, and these extracts were then by using the half dilution method, these extracts were we obtained the required concentration series. Here, the negative control we used 5% ethanol, and the positive control is 5% DMSO. So during the assay, around 10 newly hatched larvae were added separately into the 5 milliliter of different concentration of the extract and the control. And after 24 hours, we counted the dead or the immotile larvae using a Sedgwick rafter, and by using the equation given in the slide, we calculated the percentage mortality. So these are some photographs of Artemia salina larvae after 24 hours exposure to different concentration of the extracts. So moving on to the results, the sponge sample collected was an encrusting sponge with short slender cylindrical digitiform processes arising at irregular interval from its surface, and it had a convolute surface. So when it comes to its skeleton, it had it was irregularly polygonal, and it had cored primary and uncored secondary fibers. So when it comes to the percentage yield, the sponge crude extract gave out the highest yield, which is 4.1%. So under qualitative chemical analysis, both extract gave out a considerable amount of terpenoids and along with trace amount of unsaturated sterols and alkaloids. But here only sponge crude extract reported sterols, saponin and alkaloids. When it comes to evaluating the toxicity, here both sponge crude extract and the hexane extract showed strong larvicidal activities against the Artemia sana larvae. Here, from the given diagram also you can see the percentage mortality increased with increasing concentration of both extract and had a strong relationship with the concentration, and the LC50 value for both extract were more or less similar, where sponge crude extract had 14.34 microgram per milliliter and the hexane extract had 14.20 microgram per milliliter. So under discussion, the toxicity of sponge crude extract, the evaluation of toxicity of sponge crude extract is very important prior to screening the bioactivities and also further developing them into a drug. Previous studies have also confirmed the toxicity from the genus Lufariella. So here the Artemia sana is a reliable in vivo model which can be used to test toxicity for many compounds, and therefore can be used as a successful replacement to higher animal models. So here both extract contained a considerable amount of terpenoids. Thus the in vivo toxicity may be due to the presence of terpenoids or due to the synergistic effect of other zoochemicals. So having strong toxic effects suggests potential anti-proliferative properties in Lufariella herdmani extract. So further investigations are underway to investigate about this and also further about their bioactive properties. So in conclusion, the sponge crude extract and the hexane extract of Lufariella herdmani reported toxicity against Artemia salina, and this reported toxicity may be due to the presence of terpenoids or synergistic effect of other zoochemicals in the extract. And finally, I would like to take this opportunity to express my sincere gratitude to the Department of Zoology, University of Sri Javadanapura, for providing necessary facility to carry out the research, and also Dr. Marco Bertolino for the assistance provided for the sponge species identification. And these are some of the references I have used, and thank you for listening.
Thank you, Ms. Kuruppuarachchi. Now the presentation is open for the question. Ms. Kuruppuarachchi, I have a question. Why you use this particular sponge? Do they have any beneficial effect on the human? Or otherwise, there is no use of doing the acute toxicity.
Yes. So this is an understudied sponge species. It was originally reported from Sri Lanka. The species from this genus is reported to having anti-inflammatory activities, strong anti-inflammatory activities. For example, Lufariella variabilis have PLA2 inhibitor, which is considered to be one of the more effective PL2 inhibitors. So before going into the before evaluating bioactivity properties, first we have to do a toxicity screening. That's why we did the toxicity screening for this understudied sponge species.
Thank you, Ms. Kuruppuarachchi. Anyway, it's better to understand the medicinal or whatever the effect before the acute toxicity, because if they doesn't show any beneficial effect, there is no point of carrying out this kind of toxicity study.
Yes, we have already carried out anti-inflammatory we have evaluated anti-inflammatory activities, and yes, the sponge species reported anti-inflammatory anti-inflammatory properties as well, besides from its toxicity. Okay, thank you.
I have a question. Why did you specifically use hexane extract? Do you have any research clue or?
Yes, besides from the crude extract, the hexane extract gave out the most, the percentage yield. That's why we wanted to.
As I can say that hexane basically gives the lipids, no, on lipids,
Yes.
lipid soluble substances. You might miss the other substances there. Water soluble fraction. Yeah. So your hexane extract contains only the lipid soluble compounds.
Yes.
Do you agree with that?
Yes.
Any more questions? So in the absence of any more questions, shall we find out the today's session? Now that we have come to the end of the paper session 1 for today. Thank you very much for listening.
Thank you all the judge panel members and session chairs, as well as the presentees who support to success this session. Now I cordially invite Dr. Krishanti Premaratna, the president of SLALAS, to deliver the token of appreciation for chairpersons. For that, first I would like to invite Dr. Mayuri Thammitiagodage to receive the token of appreciation. Thank you, ma'am. Now I invited Dr. Ureshini Karunaratna to receive the token of appreciation from Dr. Krishanti. Thank you both. Next, we move to the very important session of the today program. Why are animal models used in scientific researches? What major scientific breakthrough have been made possible? These are very familiar areas we have to deal while we are working with the animal laboratory science. So this is time to share his valuable thoughts regarding that. So I cordially invite our guest speaker, one of the member of our judge panel, Dr. Anurag Jayasuriya, to deliver his speech. Same that time. So I invited Dr. Gihani Jayavira, Department of Basic Science, Faculty of Science, Kothalawala Defense University, to read the citations.
Thank you very much. It's a great honor to introduce the next guest speaker, who is also a teacher of mine in my undergraduate career. Dr. Anurag Jayasuriya obtained his BVSc from University of Peradeniya in 1994, and he completed his PhD at RMIT University, Australia in 2005, and his postdoc at Memorial University, Canada in 2009. He has been serving as a senior lecturer of Faculty of Veterinary Medicine and Animal Science, University of Peradeniya since year 2005. He has many publications in peer-reviewed index journals and have 5 patents by the Intellectual Property Office of Sri Lanka. He has received several awards, including the Presidential Award for Research, Sri Lanka, for his scholarly work. Sir, it's a great honor to invite you on the stage.
Yes, salah sekian. Remove my mask because I thought it is comfortable for me. So thank you very much for inviting me for this talk. And I would like to thank everybody for inviting me for this one. And topic of this lecture is actually basically focused on some of the studies that I have been doing since my PhD work in Australia, Melbourne. So as you can see, it is basically it is very vague topic though. I hope you can come to a certain level of understanding once I finish my lecture or the talk. So this is a new role of an all molecule. There are so many molecules which have been identified with new roles. The all molecules, the new roles. That is why we are doing research. So we are going to identify new roles. That is because of our curiosity only. We actually invent new roles of some molecules that we already know. So because of that, actually, the people, the human being, actually, as well as animal, are benefited because sometimes we will be able to use those molecules, the actions, to benefit of the living being. So in this talk, I'm going to show you this new role of all molecules. How I discovered using an animal model. So before going to talk, I will just give an introduction of the biological system, which is very familiar to physiologists, hopefully. And this is renin-angiotensin system, but it is extensively used by clinicians to control blood pressure. Because you know, in our day-to-day life, we use a lot of inhibitors on this system. For example, I use Rephase. It's a drug which actually inhibit AT1 receptor, which will actually inhibit the action of angiotensin II of the renin-angiotensin system on the blood vessels, which will actually limit the constriction and regulate the blood pressure. And some people use perindopril, which is also a drug which actually inhibit angiotensin converting enzyme, which actually convert angiotensin 1 to angiotensin 2. So now if you look at this system or the flow, so now you can see angiotensin 2 is the most bioactive compound in this renin-angiotensin system. As you all know, the renin-angiotensin system is ubiquitous. I mean, it is present in most of the tissues, liver, so in the body. But it basically, it originates from as we know, the protein angiotensinogen is basically originated from the liver, but it conversion occurs in different locations, for example, kidney and lungs. So actions are occurring in different tissues. So if you look at this chart, so you can see, so it is enigma, it is unknown. AT2 receptor activity is still unknown. The people are looking at that. What are the actions? So new roles are being invented. And angiotensin 1 to 7 actions are still known. It's still unknown. As I can remember those days, they are looking for angiotensin 1 to 7 actions on brain, the memory, and all these actions. So it is very interesting that the people are now discovering the fact that renin-angiotensin system is present, the whole system, the complete system is present in the adipose tissue. So that means the adipocyte. So that means the angiotensinogen and angiotensin 1, angiotensin 2, all these molecules are generated. Even the precursor angiotensinogen protein is also present in the adipocytes. So that means in our body fat, it is like an endocrine organ. Now it is not like just body fat. The adipose tissue is an endocrine organ. So probably in future lectures, probably we can include adipocyte or adipose tissue also as an endocrine organ, but we are not focusing that fact current, in the lectures and all. But sometimes we tell students that, okay, and the adipose tissue also generates some hormone-like substances, so hormones. So in this graph or this figure, you can see the whole angiotensin and renin-angiotensin system is present in the adipocytes. Okay, now I'll jump into the the incidence in the society side. So the body fat accumulation or the obesity is one of the major issues, even in animals, right? So it causes metabolic syndrome, diabetes, or like hypertension, all these things, or even cancer. So therefore, people are doing a lot of work to control the body fat accretion or body fat accumulation. So we are doing exercises, we do like dieting, all these things. So, but if you can find something that we can control this accretion or the accumulation of body fat in the body while we are doing other work in the routine work, normal way. So that would be very interesting, right? So we have to do a lot of hard work, do exercises in the evening, do a lot of involuntarily. So actually, without any, we don't actually like to control our diet. We like to eat more and more, but only thing is the consequences are very dangerous. Therefore, we have to do that. So therefore, as you can see this slide, this is not even pleasant to see, but you can see the body fat accretion or in the animal model, you can see, especially in the retroperitoneal area, in a male rat or male mice, you can see in the epididymal fat area. So in using animal models, we can study the body fat accretion and action of some of the chemicals on the body fat accretion. So in that case, we have to locate the most prominent areas where the body fat accumulated. So if you just look at the data, in Sri Lankan data, the severity of this problem is very visible in this slide. So you can see overweight and obesity in Sri Lanka. This data shows the incidence in women. The very high percentage of women are affected with this obesity. So therefore, now you understand the importance of looking some, therapeutic therapeutic methods to control this problem, rather than, telling that you should not eat, you do exercises. So as scientists, of course, we have to look at some possible ways to control this problem, or maybe we can invent some treatments. Then we can introduce that treatment to the community, medical community, veterinarians, medical doctors, probably they can use this. So I will show you very interesting finding that I was able to unravel during my PhD work without doing much, like expenses or whatever. Okay. So in that case, so if you want to visualize or if you want to measure the body fat without harming the animal, so probably you have to scan. So in that case, I used two techniques in my studies. One is the DEXA, which is actually used for human subjects, especially to measure the bone mineral density. So bone mineral density measurement is very, very common one, which is done using this DEXA machine, DEXA technique. But I use this DEXA technique to measure the body fat. Now we straight away it gives the percentage of body fat and the lean mass. So basically, this is meant for human, but I was so smart enough to use this one for animals as well in a very well-developed hospital. So other technique that I used was to visualize body fat. I used MRI. MRI, in the lab that I will show you where I work, But in that lab, fortunately, we had a MRI machine. So animal MRI, so using that one, we were able to scan the animal and visualize the distribution of fat. So now if you look at this picture, you can see the white area is fat. So it's very clearly see the fat accumulation in different areas of the body. So those are the main techniques that I used in my studies in this kind of work. So anyway, before I go into the actual talk, I just want to introduce my supervisors. One is Professor Andrew Sinclair. He's actually a lipid scientist and biochemist, and he was in the editorial board of Lipids. And the other one is Richard Weisinger, Professor. Richard Weisinger, he's a physiologist. He's the one actually worked with me very closely, and he was working on animal models most of the time. And so the Richard's area was basically ingestive behavior and blood pressure regulation, sodium appetite, and those kind of work. So he used a lot of animal models. He did a lot of surgeries to induce so many things in the brain and all those things. So he used number of animal models having different actions of angiotensin activity. So renin-angiotensin activity, angiotensin 2 activity. So he used transgenic animals, so knockout animals. So his focus was different. So my focus was actually different. So one day I thought that some animals, they have different behavior, and according to literature, I found there are some research clues that these animals might have different kind of phenotypic characteristics. So based on that, I made some observations. So in that case, I did a small work or the study using one animal models Richard used, that was angiotensin converting enzyme knockout mice. That means that in those mice, angiotensin converting enzyme has been deleted. So no action of angiotensin converting enzyme. So therefore, so now you would see that if this enzyme is knocked out, so the angiotensin 2 enzyme activity will be very less or minimum. So therefore, these actions also will be minimum. So therefore, I thought that, well, I will look at these animals and evaluate some phenotypical characteristics or the physiological behavior or biochemical features. So as I mentioned earlier, I had all techniques, the most important techniques in our lab. So I had MRI, and in the vicinity, I had the DEXA machine also. So then I used the 2 animal models. The one animal model, one is knockout mice, and the other one is wild type in my study. And well, because in those days, there are some initial discussions in the literature that angiotensin 2 has some kind of effect on the adipose tissue development. So people haven't done much work though. So therefore, I found that link, and I was so curious to see how the body fat is accumulated in this knockout mice, because if the people say, all right, angiotensin 2 has an effect on body fat accretion, so well, what will happen to the knockout mice? Then what I scanned animal, then I found, interestingly, I found in all sections, the body fat is the amount of body fat is minimum in all sections as compared with the wild type or the unknockout. So the wild type mice, okay? So these are the numerical values of DEXA results. So now even body fat also, body weight also reduced in the knockout mice as compared to the wild type. And when I analyzed the data of body fat, there was a significant reduction of body fat. Even in the MRI images, we can see that effect, but the DEXA results gave me the numerical values. The numerical values showed that knockout mice had 50% reduction of body fat, right? So that means in the knockout mice, because of the lack of the active or the most bioactive compound angiotensin 2, body fat accumulation or body fat accretion has significantly reduced. That means angiotensin 2 inhibitors, wherever it affects like perindopril or the ACE inhibitors or the drugs that act on the receptor level, would be important as compounds to inhibit the body fat accumulation, or anti-obese drugs can be used as anti-obese drugs. So because of this interesting finding, we actually evaluated the activities of these two animal models. So not same animal model, different genotypes, right? Knockout mice and the wild type. So we didn't find any significant difference between the activity level, that means distance or general locomotor activity. That means we use running wheel experiment for evaluating this activity level of these two genotypes, but there was no difference. That means the body fat reduction was not related to their activity. And even their metabolic rate was not different. Metabolic rate was also not different. And the glucose, then I suggested Richard, shall we do a glucose tolerance test? So when we did the glucose tolerance test, we found that the angiotensin converting enzyme knockout mice has a significant reduction of significant improvement of the glucose tolerance as compared with the wild type. So that means, as I have already informed or showed you, angiotensin 2, the most bioactive compound of the renin-angiotensin system, has different functions in the body. That means one is the body fat accretion, as I mentioned in my first slide. So I showed you it actually affects on the glucose transportation also, right? It may adversely affect the glucose transportation. That means if our body system has increased level of angiotensin 2, we usually use a lot of medications to inhibit this renin-angiotensin system. So as I mentioned, we use perindopril. It's a very common antihypertensive drug, which will inhibit the angiotensin converting enzyme activity. And the losartan is also very common antihypertensive drug, which actually inhibit the action of angiotensin 2 on the cells. So if we knock out the gene and if you reduce or eliminate the action of angiotensin 2, probably the animals may get a lot of beneficial effects. Or for example, so this is a knockout model, but I'm not telling you that we have to knock out the whole gene, but we can certain level, we can inhibit the activity. So we can get that beneficial effect. For example, if you inhibit this activity for a certain level, we can control the body fat accumulation, which is very important, significant and important finding that we can actually disseminate to the medical community. So overall, these results were published in one of the best scientific journals in the world, PNAS, and they disseminated this knowledge all over the world once we discovered this. And our model was that indeed in our study, we found the knockout mice and the wild type, they didn't show any difference in the food intake. The food intake was same and digestive efficiency was same. I'm not going to show all the results though. But body fat composition, as I showed you, is significantly reduced, and abdominal adiposity was significantly reduced, but the lean mass was not different. That means that the muscle mass was not different. That is also very significant effect. Body fat is reduced, no muscle mass reduction. So that is one of the things that we are expecting, right? And the locomotor activity was not different. The whole body temperature, whole body energy expenditure was different increase in the ACE inhibitory activity. And also, we found interesting, even though I didn't show you the results, some enzyme activity. For example, lipoprotein lipase activity is increased in the knockout mice. And carnitine palmitoyl transferase activity also increased in the knockout mice. So that will induce the fatty acid beta oxidation. So, well, once we did the breakthrough, it was disseminated all over the world. There is a very high impact of our research on the scientific community. So, well, Science Journal, Science Signaling Journal, we selected our result or our finding as an editor's choice. And they also disseminated this finding all over. Science is one of the best journals, number 2, of course, in the world in terms of science. So, and also the mass media, even BBC, actually disseminated this finding all over the world, right? They said scientists make weight loss claim, but they were just a little bit curious, and they were a little bit pessimistic because this is a drug which is used for hypertension. If you give a anti-hypertensive drug at a very high dose, there may be side effects. That is one of the concerns that we also had, because we have to determine the correct optimum dose to control the adiposity using this inhibitors on the renin-angiotensin system. We can get perindopril, very high dose, probably we can reduce the body fat. We can get losartan, very high dose, and we can reduce the body fat. This is not the case, right? So if you get that, it has a generalized effect. I mean, it affects all over the body. So that might give some adverse effect. That was one of the concerns that they raised after these news articles and publications. Well, this finding or the action of angiotensin II on body fat accumulation or body fat or the obesity was confirmed in another animal model that I used, that is REN2 animal model, REN2 transgenic rat, which is also an animal model which has extra renin gene. That extra renin gene means opposite to the other one, it has more angiotensin II. More angiotensin II means they must have more fat. So in my studies, I found their body weight. This is a Sprague-Dawley rat. Sprague-Dawley rats are normal rats. These are the REN2 rats without perindopril, without ACE inhibitors. There was no difference in the body weight, but there was a significant difference in the body fat. That means angiotensin II is the culprit here. It is the one which actually induced the body fat accumulation, right? So then we gave perindopril inhibitor to this REN2 high fat or the obese animal. Then we found there was a significant reduction of body fat. That means that is confirmed. That means ACE inhibitors or AT1 receptor inhibitors can be effectively used to control the body fat accumulation. Right. So what is the take-home message? Take-home message is the presence of complete renin-angiotensin system in the adipose tissue. Okay. So even though we just tell adipose tissue body fat, but it is an endocrine organ. Not only angiotensin system, there are a number of bioactive compounds have been discovered in the adipose tissue. And also angiotensin II plays a key role in the body fat accumulation. So now the next one is inhibitors on the renin-angiotensin system can be used to control the body fat accumulation or the obesity. So what is the most important thing that I want to tell you, right? In the nature, because a lot of people are doing on biochemicals or phytochemicals. There are a number of phytochemicals which are having actions, inhibitory actions on the renin-angiotensin system. That means there are so many ACE inhibitors in green tea or the black tea. So probably we can explore this possibility of using the high concentration of some phytochemicals present in the green tea or black tea to use to control the body fat accumulation obesity in future. So we have to do some studies, but we can use so many animal models, as I said. So there are animal models which have been developed focusing on these studies. Probably we can use those in future and explore some new ways to control this menace in the society. Thank you for listening. If you have any queries, you can answer. Yeah.
I am wondering Now the RAS, as you mentioned,
Yeah.
we used to call RAM, renin-angiotensin-aldosterone mechanism.
Yeah.
Usually it is activated when there is extracellular fluid volume depletion resulting pressure
Yeah.
depletion.
Basically for vasoconstriction.
Vasoconstriction and
Yep.
as well as for the sodium and water
Yeah,
reabsorption.
retention.
Yes. Now, how do you relate this one? Now, without any reduction in the volume and the blood pressure,
Mhm.
there is no RAM activation or
Mhm.
RAS activation. How do you relate this? How is there a connection? So what have you?
Well, that is a deviation of physiology now. So, but if you just look at some people who are having hypertension, so most of the doctors, they prescribe the drugs which will inhibit the RAS activity. So that means even they are not looking at the volume or whatever, though they just focus on the RAS activity and they just, for example, there are some drugs for propranolol like drugs, which are meant for the hypertension, which will reduce the heart rate and the contractile property of heart. But most of the doctors, they don't recommend even. So they actually recommend the RAS activity, even without, the body volume, body water or the body fluid retention. So some people might have even highly expressed renin-angiotensin system, which are expressing the high fat accumulation in the body. Because I think later on, after we publish this research, this has been cited by 199, I think 200 papers by now. I just got this data from the Google Scholar. So that means they have done some studies in the population studies. They have found in some population, some people have overexpressed angiotensinogen and other overexpressed proteins in their genome. Overexpressed genes in the genome, and also the protein levels in the body. So therefore, not only as a compensatory mechanism for the fluid loss, some people have overexpressed or the changes in the genome also, which will predispose them to accumulate the body fat. So therefore, if we can find something which we can inhibit this one or the control this one, so probably we can explore some therapeutic methods to control this problem. Okay, thank you very much for listening.
Thank you so much for your valuable speech, sir. I would like to invite Dr. Krishanti Premaratne to deliver the token of appreciation to Dr. Anura Jayawardena. Thank you both. Now it's time to have a small break and all are invited to enjoy the tea break.
Check. 1 takes 1 check.
Good afternoon, all. We will next move to our next session. I cordially invite our session chairs, Dr. Sugandhika Suresh, on behalf of University of Jayawardhanapura, and Dr. Chamini K.D. Silva from University of Kelaniya to take over the free paper session, part number 2. And I also give my remind to our judge panel, Dr. Anura Jayasuriya, as well as the Professor Naina Vijayawardena and Professor Charita L. Gunasekara to participate in the event.
I hope the presenters are ready. Let me just mention the ground rules of the session. Each one of you will have 10 minutes of presentation, and the first bell will ring at 8 minutes, and that will be just one bell, and at 10 minutes there will be 2 bells that ring. So at that point you have to stop, and there will be a 5-minute question and answer session for each presenter. And we have 5 presenters in this session, and as we understand, one is going to join online. And we would like to wish you all the very best, and hope all of you will do wonderful presentations. Over to you, Chamini.
Good morning, everyone. So I would like to invite our first presenter of free paper session 2. The title of the presentation is Comparison of Macrophages and Glial Fibrillary Acidic Protein Expression in TAA-induced acute rat liver injury. Authors are Tennako H, Vijay Sundara KK, Golber HM, Tanaka M, Izawa T, Kuamura M, and Yamata J. Presenter is Vijay Sundara KK.
Ladies and gentlemen, so this study is on comparison of macrophages and glial fibrillary acidic protein expression in thiacetamide-induced acute rat liver injury. So, of course, again, this is on liver, and fibrosis is the final pathway for healing. So, when you look at the liver, so there would be, when there is some sort of an injury, so there would be certain acute changes, which is known as early fibrosis. And if the injury is removed, so then the normal liver will be restored. But if this injury persists, so that would be leading into cirrhosis. So in either cases, in early fibrosis or cirrhosis, so there are 2 cells that are playing a major role, that are the macrophages, and the other important cell is myofibroblast. So macrophages, of course, they are mainly involved in inflammation, modulating inflammation. At the same time, it has been found they would be inducing the other type of cell, that is the myofibroblast. So when you look at myofibroblast, they are the important cell in liver injury. So they are the key fibrogenic cells. So they are the cells that are producing the extracellular matrix like collagens, laminin, elastins in fibrosis. And they are again mesenchymal cells. They are in between the fibroblast, that is one extreme, and the smooth muscle cells, the origin. So normally, if you look at the myofibroblast, they're in the intermediate stage. So normally it is thought, it is speculated that they would be originating from different origins, from hepatic stellate cells, fibroblast, smooth muscle cells, epithelial cells, and sometimes they might be migrating from bone marrow cells. And normally and it is thought most of these myofibroblast, they would be originating from the hepatic stellate cells, especially in the liver injury, and they would be inducing fibrosis. So one of the major factors that coordinate myofibroblast activation by macrophages would be TGF-beta, transforming growth factor beta-1, and the other one would be the platelet-derived growth factor. So those 2 would be coordinating the activation of myofibroblast. So our target protein is glial fibrillary acidic protein, which is a type 3 intermediate filament protein that constitutes the cytoskeleton of myofibroblast. And many studies they have done, and they have found this particular protein. It is found in brain, specifically in brain, in astrocytes, and mainly in astrocytes. So however, we found actually this GFAP expressed in liver hepatic stellate cells. So hepatic stellate cells, they are the precursors of myofibroblast, and the main function of hepatic stellate cells would be vitamin A storage in the liver. So however, the role of GFAP and its activation by macrophages, it's not yet clearly defined. So again, so in order to identify the role of macrophages and the association between macrophages and GFAP, we induced thioacetamide-induced liver injury. And this is our objective, to observe the association between macrophages and GFAP expressing myofibroblast in thioacetamide-induced rat liver injury. So for that purpose, of course, we used male F344 rats, 6 weeks old, and thioacetamide was injected once at a higher dose of 300 milligram per kilogram body weight. For comparison, we used control rats. So control rats were injected with normal saline and equivalent volume, and samples were collected on day 1, day 2, day 3, day 5, day 7, and day 10. Again, liver sections were taken on these days and fixed in 10% neutral buffered formalin and Sambonis for histopathology and immunohistochemistry. So histopathology was done for hematoxylin and eosine and azan stain. Then single immunolabeling was done for glial fibrillary acidic protein and CD68 and CD163. So these are two different populations of macrophages. So exudate macrophages and resident macrophages. Exudate macrophages, of course, they migrate from the circulation when there is an injury, whereas resident macrophages, they are living and they are staying inside the liver like Kupffer cells. Then real-time RT-PCR was done for TGF-beta1, PDGF, and GFAP. So this shows our results. So again, this is control livers. They are showing normal hepatic architecture. So day 1, around the centrilobular regions, I forgot to tell you, this thioacetamide mainly induces injury around the centrilobular region. So it causes initiated injury around the centrilobular region where we observed necrosis of hepatocytes. Then day 2, the maximum injury was observed. At the same time, some myofibroblast proliferation was observed, but day 3, the highest number of myofibroblast was seen. And day 5, of course, the healing process was completed. And by day 7, the normal hepatic architecture was restored. So it was compared with azan for fibrosis. So again, we observed the maximum fibrosis on day 3. So this is immunohistochemistry for GFAP, and control livers GFAP expression was only seen in the hepatic stellate cells. Mainly they are lying in the sinusoids, and they are mainly storing vitamin A. Then day 2, their number was increased, and now they are transforming into myofibroblast. And day 3, the maximum number of GFAP expressing myofibroblast were observed. And by day 7, their number was reduced. And this shows the TGF-beta1 and the platelet-derived growth factor B relative expressions, the mRNA expressions. So TGF-beta1, so it was showing a high expression, significantly increased expression on all days. And the maximum expression was shown on day 3, whereas platelet-derived growth factor beta, there was a significant increase on day 1 and day 2. So this is immunohistochemistry for macrophage markers. So again, this is CD68 for the exudate macrophage. So they were mainly seen around the centrilobular region, whereas day 2, the maximum number of macrophages were observed. Then CD163 exudative type of macrophages. So their number was less in the control livers, but day 2, there was a higher number, mainly around the injured tissue. So in order to compare these, so we checked the expression of GFAP and macrophage cell count, and we found that there is a positive correlation, especially at the initial stages of the myofibroblast activation. So with these results, of course, we came into the discussion so that there is a possible positive correlation between macrophages and GFAP expression especially at the initial phase of liver injury. And macrophages activate GFA expressing myofibroblasts in thioestimate-induced acute rat liver injury. This is our references, and thank you so much.
Thank you for that interesting presentation. Now the paper is open for questions and answers.
So US study basically important for studying
That's right, sir. Pathogenesis.
pathogenesis
to be
of
precise.
diseases, no? So in terms of implications, clinical implication, you think any link between your findings and?
Of course, yeah. I mean, with this one, we saw that there's a correlation between macrophages and activation of myofibroblasts. So myofibroblasts are the ones that deposit the collagen and cause cirrhosis or liver injury. So if we can reduce the number of macrophages, probably it would be reduce the fibrosis. So it has been found, there's another study that we have done using clodonate. So clodonate, of course, they kill macrophages. And we have found that when clodonate was given in the form of liposomes actually. So like when it is given to the liposomes, of course, macrophage ingest and macrophages die. And of course, the fibrosis would be going down. Yeah, but as I'm thinking, I mean, we are just focusing on the liver, but the depletion of macrophages might be not good for other organs as well. So we have to do more studies to find out effect on other organs.
Dr. Kavindra.
Yes, madam.
This is me again, another nice study. Thank you for the nice presentation. I just wonder how many animals you used in this experiment?
Sorry, I forgot to tell you actually. Yeah, thank you so much. I used 4 rats per group.
Okay, right. Was this done in a Sri Lankan lab?
No, this is actually done in Japanese lab.
Okay, right. That's what I wanted to know.
Avinda, may I have ask a question? Is that number is enough to get a result? Actually, we can conclude?
Yes, of course, we could not. We, of course, to get the number of animals, we used resource, some alternative method there.
Resource equation?
That's right, resource equation.
You use the resource equation to calculate the number
Of animals, right? So the minimum number was 4.
Okay.
Any more questions?
I have one question.
Yes, madam.
Yes, yeah, this is Ravsharita. Yes, actually, like when you induce this cirrhosis using thiazidamide, like how long does the effect last? So do you have to induce it like sequentially? Like, do you have to inject thiazidamide to the rats often, or like one injection is enough to have the effect for a long time? Is it a chronic effect or acute effect?
Actually, madam, induced to cirrhosis, you have to repeatedly inject it. Otherwise, if you stop, it will be revert back to normal liver. But this particular study is actually on acute injury. So we gave only one injection. And within 10 days, of course, it will be revert back to normal liver.
Normal liver, okay. Thank you.
I guess there are no more questions. So in the absence of any other questions, let us thank the presenter, Dr. Vijay Sundara, for the interesting presentation.
Thank you.
We'll move on to our second presentation. The presentation is on analysis of pain and distress during blood collection procedure from the middle ear artery of rabbits. Authors are Ratnadivakara H, Thamitiyagodage MG, Karunakaran R, Gunatilaka M, Thibault JC, and clicker F. The presentation will be presented by Ratna Divakara H.
Good morning, ladies and gentlemen. The topic of my presentation is analysis of pain and distress during blood collection procedure from the middle ear artery of rabbits. This middle ear artery bleeding is a frequent procedure used in laboratory rabbits, and it allows you to collect a quite large volume of blood in one bleeding, even though it causes some kind of potential pain and distress to the rabbits. So researchers have adapted several methods to reduce this pain. Two of those are using topical anesthesia and using aseptic techniques. Based on the physiological and behavioral criteria observations, there are several things that we can observe in rabbits to identify the pain in them. Those are like anxiousness, hiding, cries, aggressive salivation, etc. And we all know that there's this scale, Grimace, known as Grimace scale, which was adapted to assess the pain using facial expressions. Even though we have a general scale to assess the pain, we don't have any specified scales for like different procedures which we can use to develop our laboratory procedures and like improve our procedures to reduce the pain which is caused to the rabbits, which is a refinement of the laboratory animals. So during this middle ear artery bleeding, we have observed that there are like other possible assessment criteria that we can incorporate to assess the level of pain and possible stress. If I tell more about the pain and like relation of pain and stress to the laboratory procedures, pain is a stressor and pain and stress can cause distress. So the biological effects that can happen due to the pain and stress can compromise the experimental results. And environmental and physiological stress, there are like both effects. Sometimes stress can increase the pain perception, sometimes it can decrease the pain perception. So the main objectives of our study was to analyze the physiological and behavioral effects on middle ear artery bleeding procedure and to develop a scale to assess the pain and stress associated with this procedure. We have used 40 New Zealand white rabbits, which was used in a study with the ethical approvals from the Faculty of Medicine and Medical Research Institute, Colombo. And the blood sampling was done in predetermined time points. First, what we did was we take the rabbit out from the cage and we let them stay on the table for some time, explore the environment, like allowing their inquisitive behavior to like express their inquisitive behavior. And then after some time, after they calm down, we restrain them with a large towel and clip the dorsal surface of the ear and apply lidocaine gel and kept for about 2 to 3 minutes and disinfected with 70% alcohol, and bleeding was performed using a 23 gauge needle. When there's situations the blood flow gets slow or obstructed due to vascular spasms, we use a gentle tap or stroke to the artery to facilitate the blood flow. These are the criteria that we assess during the procedure. We measure the Grimace scale prior to the procedure to see the basal level, if there's any basal level of pain before starting the procedure. And we calculate average number of needle pricks per ear, and also the presence or absence of hematomas and the size of hematoma. The recordings were done by a third person who is not involved directly to the procedure. And each item or the criteria was individually given an ordinal scale and collectively a scale of 4 to 30. And it was converted to 1 to 10 scale, where 1 being 0 pain and distress and 10 being severe pain. And also we have done this evaluation of Cronbach's alpha value for the internal consistency of item scales. This Cronbach's alpha was used to measure the consistency of these kind of scales, like the questions and items in a scale. It shows how closely related those items are. In this table, the items of our scale is summarized. Here you can see how each of the items was given a scale based on the results. in this table, as you can see that we haven't observed any significant difference or gradual reduction like towards the continuation of the procedure, but we have seen like when we go towards the latter stage of the procedures, the hematoma percentage and the pain, basal level pain of the rabbits and the physical reactions are kind of reducing, but it's not statistical or like gradual reduction. And this pain level, which was observed at day 0, we started this procedure at the rabbits were 3 months old, very young age. And it's the first time that they were handled in that kind of a procedure. We wrap them and like do those kind of procedures. So this basal level day 0 pain, which was observed like 20%, would be due to the handling and the first time exposing to the procedure. As I've told you, the reduction of hematomas happen due to the behavioral response to the procedure when they pull the ear and when they struggle or something like that. So we have seen the reduction of behavioral response towards the towards the latter days of the procedures, where gap between the two bleedings were increasing, or as the recovery period is increased. So this kind of shows that the level of pain and the related stress is influenced by the history of impignate stress during the initial days, and the memory and the history of previous stress will be like reduced towards when the gap between two bleeding is increased. This is the Conbrax alpha value for each of the items for each of the days. And in the Conbrax alpha values, we say if the value is over 0.6, it is acceptable. And if it's getting bigger, like the value is closer to 9, it's excellent. The relation between the items are really excellent and have a close relation. These are the values that we have received. And we can say the 4-item scale is acceptable. And also we have measured the internal consistency between the items collectively for all sampling days. And it is also more than 0.6, which is acceptable. With the continuation of the procedure and the aging of the rabbits, we can say that they get familiarized to the procedure and the level of stress-related behaviors reduced. And also during the study, we have seen that applying anesthetic gel 2 to 3 minutes prior to the procedure was effective in reducing the level of pain and reaction. Strict adherence to the sterility throughout the procedure resulted the hematomas on the ears get healed within 4 to 5 days. Our preliminary finding shows that the behavioral response was effective measure for assessment of the pain related to a specific procedure, but apart from the behavioral findings like responses and all, we can correlate other biological responses as well as like endocrine and other kind of like measurements, like quantitative measurements to incorporate into this scale. And then with more data, we can assess do a more assessment and reliability and consistency of the work. Thank you.
Thank you, Dr. Ratna Divakara. It was a very interesting presentation. Now it's open for discussion. Any questions from the audience or the judges?
So you have basically done behavioral responses.
Yes, yes.
So do you have any idea in future to do some, like you have mentioned in abstract,
Yes.
to assess the improvement using some hormonal assays?
Yes, we can do like the stress hormone levels and blood pressure evaluation and those kind of things. And also the pain assessment at the end of the procedure, we couldn't perform because there's only one person observing and recording. If we have the facility to do like video recording or something like that, we can assess more accurately like how the Grimmett scale will vary towards the end of the procedure.
Clearly that, you know, once the animals acclimatize to the condition,
yes,
probably it will make some bias to the results, no?
Yes. Like when we go to the animal house also, like when they know the procedure and all, there can be bias as well. Yes.
Any other questions?
I just want to say actually, it was a very interesting presentation because we as researchers always use animal models for various research, and this is a very important consideration and aspect that you have studied. So I just wanted to comment on that.
Thank you, madam.
Receive the ethical clearance from
Yes,
the university?
yes.
In the absence of any other questions, let's thank Dr. Ratna Divakara for her interesting presentation.
Thank you.
Let's move on to our third presentation of the session. The presentation is on effect of partial substitution of rice polish with de-oiled rice polish as energy supplements in broiler diet. Authors are Pereira DPRA and Pereira Jayavira BBA and Manopriya S. The presentation will be done by Manopriya S.
Warm greeting to all. Today I'm going to present about my undergraduate research, which is on effect of partial substitution of rice polish with de-oiled rice polish as energy supplement in broiler diet. As we all know, in Sri Lanka, 80% of the population is depending on broiler meat as an animal protein source. So with increasing the population and demanding towards the broiler meat, we have to produce more. At the same time, there is a big, huge problem actually. With increasing demand, the farmers are decreasing. The interest of farmers are decreasing towards the poultry production. The major reason was increasing cost of production. So what may be the reasons? So when we going in Sri Lanka, poultry industry is 70% of the livestock sector. So when we think about this cost of production in broiler, nearly 59, around 60% of the total cost of production is feed cost. So if we want to cut down the cost of production, we have to think about the feed cost. So there are several constraints which inhibit us to reduce the cost of production. First one is high cost of formulated feed. Since most of them are small scale farmers, they are using formulated feed which are already available in the market. Second one is, even though a farmer is willing to produce their own feed, the scarcity of the feed ingredients in the market. Most of the soya and maize, those are scarcity. Because of this scarcity, and we are depending on mostly in the imported sources, there is an increasing price in feed ingredients. So what we can do to reduce this? So we can go for an alternative feed ingredient rather than using the imported or high cost feed ingredient. So de-oiled rice polish is one of the substitute. So as you all know, the rice polish is one of the major energy source which are using in most of the animal feed, such as poultry and cow and everything dairy farming we are using. But this rice polish can be used in another effective way, that to produce rice oil. So when we are producing rice oil, there is another byproduct coming out, de-oiled rice polish after extracting the oil. So we are assuming this de-oiled rice polish has better nutritive value, not same as rice polish because we are removing the oil part, but it has some amount of nutritive value, and also it has a high potential to utilize as an energy supplement for rather than using a traditional these energy supplements. So the major objective of this study is to identify the suitability and the optimum inclusion rate of de-oiled rice polish to formulate broiler diet for better broiler performance. Initially, proximate analysis for the de-oiled rice polish sample has been carried out. After that, there were several feed ingredients were selected to formulate the broiler feed. Here I have shown the feed ingredients. Then broiler starter and finisher rations were formulated using ration builder software, and all these rations were prepared. Here, one control and three treatmental diet was prepared. Control diet is fully with the rice polish, and all these other three treatmental diet was prepared by de-oiled rice polish by adding 10, 20, and 25% rather than using the rice polish. Next, the diet has been prepared. Then finally, feeding trial was conducted. Finally, all these data were analyzed using SPSS 16 and the Excel. Here you can see the starter ration for the broiler. This starter ration was feed from day 1 to day 20. All these diet were more or less equally maintained with the energy, metabolizable energy, and the crude protein level. And here we can see the finisher ration, which I have used for the broiler diet. Here clearly we can see I have increased 10% and 20 and 25% gradually with the de-oiled rice polish without rice polish. Fine. The total number of the birds for the feeding trial has been calculated using resource calculation, and altogether 96 day-old chicks were selected, and it has been grouped into 4 treatmental groups with 3 replicates, 8 birds per replicate. Then from 0 to 21 days, starter ration has been feeded. After that, 22 to 42nd day, finisher ration has been feeded. Finally, 3 birds per each replicate has been slaughtered, and it has been analyzed for the meat quality, especially carcass yield percentage and main 8 cuts of meat, and also giblets also been analyzed. From these measurements, I have calculated the feed consumption rate, body weight measurements, and live weight, carcass recovery percentage, and carcass quality and carcass traits, and also giblets. Here, these are the common equation that I have used. It has been already accepted internationally. Feed consumption ratio, broiler performance index, and broiler efficiency index. Here, the broiler performance index only shows what is the final yield, what is the efficiency of that bird. But the broiler efficiency index shows how it's viable. As a commercial level, how it's viable, because it has with the feed cost and other relative costs. So from this, we can identify whether it's commercially viable or not. So let me move on to the results. Here we can see the proximate analysis of de-oiled rice polish. The dry matter content is 93.43. The energy is 1800 kilocalorie per kilogram. In the initial rice polish before removing the oil, it's nearly 2800. Due to the removal of most of the oil part, the energy has been reduced up to 1800, but still we can utilize it as an energy supplement up to some inclusion rate. So crude protein is still remain as 14. It little bit increased because that fat part is reduced comparatively. The protein and fiber part is increasing. So here we can see the variation in the feed intake and final body weight, feed consumption ratio, and broiler performance index and broiler efficiency index. Normally, the recommended value for the feed intake for a bird is 3,500 grams. Here we can see all these birds, most of the, without any significant deviation, it's relatively combination with the recommended value. And final body weight, we have expected 2 kilograms, but all the birds were really reach up to 2 kilograms. Some of the birds nearly gone beyond the 2,200 kilograms. And the feed consumption ratio, it's also nearly the same with the recommended level 1.6, and broiler performance index, higher the value, higher than 1, it means it's showing good broiler performance index, but it's nearly, with the reference and the literature, it's nearly between the 35 to 50. We can say we can effectively use this as a economically effective method. Here we can see the daily weight gain of the birds during the research period. So with the control, when we are comparing with the control, there is no any much deviation. But in the 12th day, there is a little bit reduction of the body weight growth. This is basically due to the climate change, because with this higher temperature and everything, birds started to consume more water. So what we have done is we have moistened the feed and we have provided, again, it became to normal. Here we can see the carcass recovery percentage. Carcass recovery percentage means after we are slaughtering the bird, we are removing all these feather part and all these skin and everything. What is the old chicken we can get? So here we can see nearly carcass recovery percentages are almost same with all these treatment without any significant deviation. And the breast yield, back muscle yield, and drumstick thigh, all these are showing nearly same value without significant difference between the treatment. But in the wing part, we can see there is little significant effect, but wing is a very small yield from a poultry chicken, so we won't bother that much as when we are thinking about the commercial level. So here we can see the cost benefit analysis. So here we can see the return on investment in this treatment. So here we can go for this as a commercially viable product rather than using a rice polish. Up to 25% of inclusion rate, we can use the de-oil rice polish rather than using rice polish as a cheaper, good quality alternative resource for a traditional energy supplement. So there are possible ways to improve these when we are considering in the commercial level, we can improve these nutritive value more than this by using a different fat sources, cheap and good quality fat sources. And here are the references that I have used, and I acknowledge these people. Thank you. Thank you for listening.
Thank you, Ms. Manopriya, for that interesting presentation. The paper is open for discussion. Any questions?
I have one question. This is Dr. Yes. So how many with how many birds did you try this study? How many samples did you use?
Yeah, altogether 96 chicks were used for this feeding trial for one treatment. Nearly 24 birds. Altogether with control, there are 4 treatments, so 96 birds. 3 replicates. For one replicate, we have taken 8 birds as a one replicate.
Okay, so basically 8 birds have been used per one feeding 26, is it?
24 birds per treatment.
Okay. So if you want to extend this into like a commercial scale or make recommendations, so do you have to do like a field study, like in a using a like actual farm, or like replacing their feeding with your alternate feeding type, and how do you plan to extend this into like a commercial level like study?
Actually, ma'am, this is also like a field study. So we have conducted this in a university research farm. So if we are, if it is a commercial level, we are going to again increase the number of birds. So again, it's going to be more benefit. So because we are using just 96 animals, so the cost of production and the maintenance cost is again increasing. So when we are thinking about the commercial level, again it will be reduced. And also we are more used to so rice is our stable food. So commonly rice polish and de-oiled rice polish are now it's commonly available in the market. So it's commercially viable.
That's good. Interesting. Thank you.
Thank you, madam.
Is it a regular procedure that you conduct in your university laboratory or the field trial?
Yes, madam. Normally feeding trial for broiler is the same way.
Right. Okay. So have you obtained ethical clearance or as you said that it's the normal procedure?
Yeah.
So is there an overall ethical coverage for your university laboratory?
Yeah, we have an ethical committee in the university level. We have.
Not that I mean. So have you obtained separate ethical clearance for this?
For this study, I didn't obtain, madam, because it's the normal procedure as a feeding trial. Normally, it's a farm level procedure.
At the end, you are killing animals.
Yeah, normally even in the for the not the research level, no, for the feeding and everything, we are normally killing the animal. So it's kind of a feeding trial. So we didn't get the ethical clearance for this particular study.
So this is a research study.
Yeah. So since it is not that much of invasive, so we didn't go for a ethical clearance.
Pardon me.
Is Dr. Prasanayana speaking online? No.
No, Professor Gandhika, I don't have any questions to ask.
I have one more question actually. So all these chickens that you have sacrificed also, have they been consumed?
That slaughtered animal we didn't consume. For other animals, we have sold out for consumption.
Same question, like, yes, if you use euthanize word, then it's not, you're not going to, the terminal of the, on the end of that animal will be like, purposely we are euthanizing. That means we need ethical clearance. If we are taking samples from the normal slaughter for the research, then no need. Purposely, if we are euthanizing it for a particular purpose, research purpose, then we need to consider the ethical clearance in the project.
Okay, thank you.
Okay, in the absence of any other questions, let's thank Ms. Manupriya for her presentation and move on. Thank you.
Let's move on to our fourth presentation of the session. The title is In Vitro Antioxidant Activities of Fresh Flowers of Clitoria ternatia. Authors are Vikramage CS, Tilakaratna WKAW, Pathiraja PARM, Tatsarani NBK, Silva N MG, Fernando MSD, and Ratnayaka WMKM. The presentation will be done by Vikramage CS.
A very good morning, all of you. I'm Sankalpa Wickramage, and my research presentation title is In Vitro Antioxidant Activities of Fresh Flowers of Clitoria ternati. So these are my research outlines. A free radical is a molecular species having an unpaired electron in the atomic orbital. So this reactive oxygen species is also one of the examples for a free radical. So when we comparing the biochemical reactions in our body, there are so many reactive oxygen species come to play a major role, such as the hydroxyl radicals, then the peroxide radicals, and the superoxide radicals. So actually what happens with these free radicals, it changes the healthy tissues into the unhealthy form. But with the presence of this antioxidant, it can prevent the cellular damages and prevent the infections. So since many plants contain these secondary metabolites, they do have this antioxidant property. So in our study, we try to focus and evaluate on the antioxidant property, especially on the plant Clitoria ternati. So Clitoria ternati is simply well known as Nilkataralu in Sinhala, and in English, it is known as the butterfly pea. So this plant, Clitoria ternati, is belong to family Fabaceae. So from the ancient time, actually, people believe that there's a significant pharmacological importance of this plant, Clitoria ternati. And that is why even today, people try to buy these plant products which contain raw material as Clitoria ternati, such as the food beverages and the coloring agents, also in cosmetics. So in our research, we try to focus and develop a facial cream. Actually, but there's a significant lack of scientific evidences in this pharmacological activity of this plant. And therefore, we try to focus and evaluate, especially on the antioxidant property of this plant. So as you can see, considering the 3R concept, before we move into an in vivo study, we have already planned and conducted in vitro antioxidant assay. So when conducting the antioxidant assay, we have used the DPPH assay by using the 2 fresh flower varieties commonly grown in Sri Lanka. The methodology was proceed like the 2 varieties of the blue flower varieties, basically the blue flower with the normal keel petal and the blue flower with the enlarged keel petal was collected from Colombo district and authenticated. After that, during the test sample preparation, actually our sample was prepared 3:1 ratio. First, the sample was weighed and then it is subjected to refluxation. After that, the filtrate was taken out, and by using that filtrate, a concentration gradient was prepared. So as you can see in this slide, the DPPH assay was conducted like this. We have used a negative control as DPPH and methanol, and a positive control as DPPH and the ascorbic acid. Also, in order to reduce the color interferences and the color changes, we have used a color blank. And also all the test samples were triplicated and the negative controls were triplicated and the color blanks were triplicated. So by using that already prepared test samples, it was kept in dark and incubated. After that, the absorbance was measured. And also by using the 50% scavenging activity, we have calculated by using the formula given here. So all the results were analyzed by using the software SPSS, and we have plotted a graph versus the mean concentration of the triplicated samples against the percentage radical scavenging activity. So now we have come to the most important part of my presentation, that is the results part. So as shown in here, both the 2 plant varieties, the blue flower with the enlarged keel petal and the blue flower with the normal keel petals, these 2 varieties show the positive results for the antioxidant assay, which is DPPH, and there's a significant oxidative potential in the blue flower with the normal keel petal as 20.09%. So this table shows the results for the antioxidant assay, which we have conducted for the ascorbic acid, and that result for the EC50 value is denoted as 26.61%. So in discussion part, the DPPH is actually a pre-radical, but with the presence of the antioxidant, this DPPH can turn into reduced DPPH form. And in here, we can see a specific color change actually from purple color into the yellow color. So within our test samples, we also see this similar color change from purple color into the yellow color. So in conclusion, we have found enough scientific evidences to prove the scientific in vitro antioxidant properties of the fresh flower 2 varieties of Clitoria ternati. And among them, the blue flower with the normal keel petal shows specific antioxidant property rather than the blue flower with the enlarged keel petal. So actually, this is only a small part of our main project, and our main project is to develop and incorporate a facial cream. Since this Clitoria ternati plant shows enough antioxidant properties, especially the blue flower with the normal keel petal, we try to incorporate that antioxidant property into the cream. And also up to now, we have developed a stable cream base. So these are some of my references. Finally, I would like to thank Faculty of Health Sciences, CINEC campus for providing all the laboratory facilities and also all the needed chemicals. And also I would like to thank Sri Lanka Association for Laboratory Animal Sciences for providing this great opportunity in science work. Thank you.
Thank you, Ms. Wickremege, for your very interesting presentation. It's now open for discussion.
Milcartol is extensively now used in making some food items. Do you think the compounds which are responsible for antioxidant activity in milcartol are resistant to heat? Because most of the food items, they are cooked. So do you think those compounds be deactivated like ascorbic acid?
Yes, we have already conducted the refluxation even within our research, and it is conducted for the 90% Celsius, and that level, the chemicals are already stabilized. So therefore, there will be no interactions with the cooking procedures.
Next plan is to, formulate a cream, right?
Yeah.
So you said you already formulate a cream base. Do you use a lot of chemicals for that?
No, so we have already, since this is a like Ayurvedic one, we have incorporated the Ayurvedic ingredients into that.
Any other questions?
This is a very simple question I'm going to ask. Do there are any other literature on this Clitoria plants in Sri Lanka or anywhere else in the world?
Yes, madam, there were some, but this one is the very first time we are conducting a research for the fresh flowers.
Okay, right, thank you.
Thank you.
And, so you mentioned that the normal plant has the highest the antioxidant property. Yeah. So what could be the difference between the normal and the other one?
The enlarged keel petal, actually the plant is showing with the petal varieties. So since we thought that there's not any comparison up to now in Sri Lankan research activity, and we try to incorporate that and do this research.
Now, it is good that you conducted an in vitro study first without going into animal studies, right? So it is replacement concept. But do you agree applying something? Yes, it is a natural thing, right? Without studying any toxic effects or adverse effects. So directly applying treatment, do you agree with that?
Yes, madam, actually we have taken the extract and we try to apply if there are any in vivo studies within the next couple of years. We try to take the extract of this plant and go with the in vivo studies.
Excuse me, very nice and interesting study. I just want to make one comment and a suggestion. Like we use Nilkatral flower as a color enhancer for ornamental fish. So and also some studies prove that it also increased their growth performance. Probably has some antioxidant activities and all. So disease resistant, I don't know whether they studied. So maybe there are a lot of compounds. So in your case, if you're going to use the whole flower extract, will there be other side effects like the color and etc.? Will it be effective as a cream?
Actually, we have up to now considering only the antioxidant property, and if there are any color effects, we try to incorporate that in the next few research activities.
Yes, that's all. So in the absence of any more questions, thank you, Ms. Wickramage, for your very interesting presentation, and hope you become successful with your product. Okay.
So we'll move on to the final presentation of the session. I think it will be delivered online. The presentation is on BR driver, a machine learning approach for predicting breast cancer driver genes. Authors are Pereira DPRA and Pereira JARP. The presentation will be delivered by Pereira DPRA.
Good morning to everyone. I think you can hear me properly. Yeah, it's okay with the sound.
Yes, we can hear you.
Yeah, thank you very much. And good morning to everyone. According to the recent studies in Sri Lanka, they have identified that over 15,000 deaths occurred due to the cancer, and most commonest cancer type is breast cancer. And in our study, we are going to address this issue by using an in silico method, the machine learning method. And the breast cancer mortality rate is high in developing countries. We know that. We have identified a reason, one of the reasons, the ability of early detection of the cancer is very low in that particular countries. And in this picture, you can see that many of the Asian countries are the mortality rate and the incident rate is greatly high in the many of the Asian countries. And this is our research problem, that there is no gold standard method for identifying breast cancer driver genes. We know that the driver genes means the genes, the mutations essential for cancer cell development. And the problem is there is no gold standard method for these breast cancer driver genes. And in this study, we are going to we are planning to develop a model to predict the high confidence breast cancer driver genes. This is our methodology. In briefly, we can go like that. Go like this. We have selected some primary data set, and by using this primary data set, we have input these primary data into different computational tools, and the results from these computational tools were recorded as the secondary data set. And when the secondary data set has produced, by using a data sampler, we have divided the secondary data set into test data and trained data. And trained data was ranked according to the correlation between the train data and the breast cancer driver genes, which is our targeted variable. And the ranking process is an iteration process. And it's like this. When the train data comes into the ranking process, first ranking, for the first iteration, we are only selecting the highest rank attributes. And for the second iteration, we are selecting first two highest rank attributes and so on. The ranking attributes were trained using different machine learning algorithms. And after that training process, the test data and trained by using these test data and training data, the classification accuracy was calculated. That's the brief end of this methodology. And in here you can see that our primary data set, it is freely downloaded from the breast TCGA Pan Cancer Atlas, and it contains this invasive carcinoma data from 1,084 patient samples. And we are using 27 computational tools, and primary data used as the input for these 27 computational tools. And the tools will calculate the effect of each gene and their variant in the primary gene set. And the results from these tools recorded as the secondary data set. In here you can see that the results will be the calculated value from each tool. These are the tools in the central section. You can see the tools, and these are the genes in, and there are some calculated values in the secondary data set. Yeah, and when we consider the results, first we have identified what iteration is produce the highest classification accuracy. You can see in here the classification accuracy, the CA means classification accuracy, and we have identified that iteration number 7 when we are using random forest machine learning algorithm, and iteration number 7 produces the highest classification accuracy. And after the classification accuracy, we have gone through the area under curves, and we have identified that iteration number 7 will produce the highest AUC values when we are using the random forest machine learning algorithm. As an example, in here you can see that iteration number 28 will produce some least values than the iteration number 7. And after that, after the AUC values, we have go through the confusion matrix, and it also in the iteration number 7 produce the least misclassified values. You can see the this value is the actual values, and the predicted values, which will be 1 is 0, and it is the least value we have produced in our study. And when we are considering the iteration number 28, it produced highest values than the iteration number 7. Yes, in our study, the TP53 is the most predicted breast cancer driver gene. And in this figure, you see that the top 10 predicted genes by using this BR driver. And especially, we have mentioned that in this study, identified 2 novel breast cancer driver genes, which are CDKN2A and NFE2L2. These are novel breast cancer driver genes. And we already know that in our study also, we have used 27 computational tools. There are a lot of computational tools have been developed to identify these cancer driver genes. But few methodologies were developed to combine these tools and increase the efficiency. And in our BR driver model, the area under curve and the classification accuracy higher values shows that it overcomes these other tools difficulties and produces the high confidence breast cancer driver gene set. These are my references. There are a lot of, but these are the main references I have. Yeah, and thank you very much for listening.
Thank you very much, Mr. Pereira. Hope you can hear us.
Yes. Yes, sure.
Yeah. And okay, thank you very much for your commitment to present the allocated presentation even when you are not well. So we appreciate that very much. And now the paper is open for discussion.
Sure.
Can I ask a question?
Yeah, sure.
Yeah, so this primary data which you have used, are they like sequences, like genomic sequences, or like the identified, like potential identified breast cancer genes in a study?
No, in the primary data set, it says like this, these are the variants happen in this data and how much time they have occurred in. As an example, one particular gene, you can say that one or ABC as a gene, how much time it will appear in that data set. That's all the things in the primary data set. It never mentioned that they are the driver gene or other things. It never mentioned in the primary data set.
Yeah, when you say like data sets, like including 1,084 patient samples, what are these patient samples?
I have chosen the breast invasive carcinomal data from the TCGA Pan Cancer Atlas.
Okay. So what kind of data available in this atlas? Is it like genomic sequence data or like?
Yeah, there are a lot of genomic sequences data, and especially with the breast cancer, it's around 10 or 12 data sets available at the TCGA Pan Cancer Atlas.
Okay.
I have chosen the highest data set number because 1084, that one is the highest data set. It's available at the TCGA.
Can you hear me?
Yeah.
Thank you for a nice presentation. I just have a very simple question.
Sure.
As you have mentioned in the first slide that most of the cases are reported in Asia, right?
Yes.
And those are deaths.
Yes.
So what do you think that why we have this kind of variations in different continents?
Yeah, as I mentioned, the early detection is very low in these middle income countries. I suggest that one is the main reason.
Yeah, and also it might be the management sometimes, the treatments and everything. It might be, I think the management also play a role. So how you have normalized those kind of bias, and in Asia we do not think that we will have much data fed into that atlas as well. So
Yes.
what do you think in the validation process, how you are going to overcome that problem? Because you have to represent entire, some kind of a statistical program. You have to incorporate to prove that this software should, run with all the available data. That's right. But the available data that you have used in the primary as the primary data,
yes,
so is it representative?
We can improve our. Actually, this is a model, not a kind of a tool, the BR driver, and we can improve the model by increasing the data set number. And in here, I have only used the highest, as I mentioned, the highest breast cancer data, but we can improve the data set number and improve our model. But you have mentioned about and Sri Lanka and other Asian countries data. Actually, they are not available in that data set, and it is a major problem in here. We haven't got any recorded data about breast cancer or other things. We haven't got any sufficient data set for these particular countries. We have a lot of patients, and there are a lot of testing done in here, but we haven't stored those data, or we haven't looked back those data, and we haven't identified the what are the similarities between those data or other things, because we haven't done the database thing first.
Thank you, Mr. Pereira. It's just for my interest. These data sets, they are freely accessible, and you all are allowed to use it for any analysis, I suppose.
Yes, ma'am. They are freely available. Yeah.
Okay, so let me thank you again for your commitment and being available online for this presentation. Thank you.
Thank you very much for giving this opportunity. Thank you.
Okay, so with that, we come to the end of the free paper session 2. And in this session, we had 5 interesting presentations spanning from, say, about the expression of GRP proteins, and then about analysis of pain and distress, and then about substitution of rice polish with de-oiled rice polish, followed by the in vitro antioxidant activity of blue pea flowers, and then finally a total deviation from the animal studies and the in vitro studies. We had this computational analysis study on VR driver genes. So a big round of applause to all the speakers who made very interesting and informative presentations. And this concludes the free paper session 2. Thank you. Let me please thank the judges also who were online and one in person for their contributions.
My thanks goes to the judges panel and the session chairs, as well as the presenters on behalf of the contribution for the last session. Now I cordially invite Dr. Krishanti Premaratna, the president of SLALAS, to deliver the token of appreciation to the chairpersons. I invited Professor Sugandhika Suresh to receive the token of appreciation. Now I invited Dr. Chamani Kanativeladi Silva to have the token of appreciation. Thank you all. Now it's time to appreciate talents. This is about the SLALAS oratorical competition for undergraduate 2021. So I cordially invite Dr. Gihani Jayaveera to give the overview of this competition.
Thank you very much. SLALAS continued its scientific and professional activities even during this pandemic time. In this year, I mean in year 2021, according to a concept by our president, Dr. Krishanti Premaratne, we introduced SLALAS oratorical competition for undergraduates. The aim of this competition was to raise the awareness on laboratory animal science among the undergraduate community and to appreciate the contribution of laboratory animal science towards the success or the progression of the biomedical sciences. We advertised the competition and we received entries from contestants. And in order to provide a basic understanding on the subject, we conducted a training workshop for the contestants. After that, contestants, they submitted their speeches through the videos. And these videos were evaluated by a panel of judges appointed by the SLALAS Executive Committee in order to select the winners. I'm really grateful for our president, Dr. Krishanti Premaratne, for introducing this concept and providing great leadership in conducting this new activity. And also to Sachini Amarasinghe, our secretary, and the panel of judges, Professor Mangala Gunatilaka, Professor Sugandhika Suresh, and Dr. Mayurita Ammitiyagadage, and all the members of SLALAS Executive Committee for their tremendous support in successfully conducting this new activity. I also would like to congratulate Ms. Mohammed Rial Fatima Litha from Khartoum International University for securing the first place in first ever SLALAS oratorical competition. Now, ladies and gentlemen, we will now witness the speech of the winner of SLALAS oratorical competition, Ms. Fatima Litha of Khartoum International University.
Animals are cut alive, abused, and tortured. The other party says the pain they suffer diminishes the pain we as humans suffer. Now the question is it gratuitous cruelty or a justifiable practice? Hello everyone, I'm Litha and my speech today is about whether animal-based research is needed for the future. These researchers, medical researchers, are carried out to discover how an organ works, how a disease affects a particular organ, and how to treat it. So researchers need efficient models to find out all of the questions, but they cannot bait humans in this risky test because they are creative, intelligent, and more sentient animals. So there are more ethical issues. In the meantime, some non-human animal species mimic the genetic feature of humans or have similar organs or express similar physiological mechanisms. And also variables can be easily controlled in them. So researchers can study the biological mechanisms and the behavior in normal or in disease condition and use those resources to develop a new cure. This is why animal testing is the fundamental requirement of medical researchers. Let's go to a few boring history now. Galen, a physician from 2nd century, is known as the father of vivisection, whose work was helpful to develop human medicine. And the great breakthrough in medicine was when William Harvey discovered the complete human blood circulation with the help of vivisection. All the medical inventions and discoveries that we are proud of now have their share of animal testing. Insulin, dialysis, cardiac pacemakers, organ transplants, vaccines such as smallpox, tetanus, polio, and there are more. Still, many people think that it's unethical to test animal, even that is going to benefit humans. Some say we can stop this as we have alternatives now. Of course, we have wide data collections, computational models, in vitro tissue culture, even in vivo testing using the chicken correlatoid membrane has become a widespread practice in many countries. But still, alternatives cannot completely replace the body system where all organs collaborate and work together. Even recently, Pfizer and Moderna officially claimed that their vaccine did not skip the animal testing. So the animal testing is still required for essential tests. But the usage can be regulated, thus abuse can be prevented. We already have 3R system, replaced with alternatives, reduce the usage of animals as much as possible, and refine. Researchers should make sure that animals won't suffer during or after tests. Nevertheless, the total abolition of animal testing will demolish the whole medical foundation and will be a threat to humanity. So my stance today is animal testing is needed for future researchers, but can be implemented with strict regulations. Thank you.
Congratulations, Ms. Mohammad Real Fatima Lita. Now it's time to raise your voice to share your thoughts.
Honorable invited speaker, Dr. Anura Jayasuriya, Honorable Judge Panel, and the entire Slalas family and the audience, good afternoon. I'm Fatima Lita, an undergraduate of Biomedical Science of Khartoum Highly Advanced Medical Technology Training Center. It's so grateful to speak in front of such an accomplished group of people about my experience on winning the Oratorical Competition 2021 organized by Sri Lanka Association for Laboratory Animal Science for undergraduates. First of all, thank Dr. Kushanti Premaratna, President of Slalas, Sachini Amarasekara, Gihani, Honorable Panel of Judges, and the Executive Committee for organizing such a competition for undergraduates like us and recognize us. Actually, one of my friends suggested the competition and we wanted to gain new experience. So after a while, we were given the topic, is animal-based research needed for the future? So although we were familiar with the scientific aspects, but as undergraduates, still we were struggling to develop the content. But fortunately and thankfully, Slalas themselves organized a training program for us and guided us through to prepare the best content. So despite the experience I gained from the process, this day, in fact, that I could meet all of you people who actually are in the positions that I'm dreaming to be one day, I must say this is a memorable day in my life. Thank you, Slalas, for giving me such an opportunity. Hope you will continue these type of activities in future too. Thank you.
Now I invited Dr. Gihani Chayveera to present the award to the winner. I invite Real Fatima to receive it. Thank you all. Next, I invite you all to have lunch refreshment at Veranda Restaurant. Before we move to the third session of today's program, we will be meet again after 1 hour at 1:30 p.m. to continue our third session of the program schedule. Please wait all to have a photograph before we leave into our lunch.
Check one.
Good afternoon, everybody. So this is time to start the final category of free paper session number 3. So I cordially invite to session chairs Professor Mangala Gunathilaka on behalf of University of Colombo, and on behalf of Uva University, I cordially invite Dr. Prasadi De Silva to take over the session and proceed up. And I cordially invite our judge panel members, Dr. Anura Jayasuriya, Professor Naina Vijayawardena, and Professor Charita L. Gunasekara to contribute their participation for the session.
Good afternoon and welcome to the free paper session 3. We have 4 presentations, 9 up for the last session. And if I remind you the ground rules, we have allocated 15 minutes, that is for both the presentation and the discussion, question and answer session. And the first will be run at 8 minutes during your presentation, and the 2 bells will be at 10 minutes time. So it is time to complete your presentation. And after that, the question discussion session. Let me introduce the first, the presentation with the title. Comparison of Colombo and North Central Province Dugwell water quality parameters by cumulative mortality percentage in zebrafish egg model. The authors are Thammithiyagodage MG, Karunakaran R, Deshapriya ABMG, Korea S, and Vanyarach D. Paper will be presented by Karunakaran R.
Good afternoon, everyone. Thank you for the introduction to Dr. Professor Mangala. Title of my presentation is Comparison of Colombo and North Central Province Dugwell water quality parameters by cumulative mortality percentage in zebrafish egg model. But under background and justification, chronic kidney disease of unknown etiology is prevalent in certain areas of Sri Lanka, such as North Central Province, Uva Province, and Northwestern Province. Majority of the people in affected areas consume Dugwell water. Heavy metals, water hardness, and other unknown in water are among suspected etiological factors. Detailed water analysis was conducted in North Central Province water, and presence of high fluoride, calcium, sodium, and different rates of other contaminants were identified. However, the reported number of CKDU cases in the Colombo district are minimum according to available literature. Majority of the people in the Colombo district consume tap water, but there are people who consume Dugwell water as well. Dugwell water from Colombo is free from toxic levels of heavy metals according to WHO guideline. Zebrafish egg model is widely used as an alternative to animal testing. It is growing popular among Sri Lankan scientists. It is popular due to its easy to maintain nature and cost effectiveness. This model is used in environmental toxicity assessment in a variety of conditions. Sri Lanka is a country with some identified environmental toxicity issues. There are different communicable and non-communicable diseases suspected due to environmental pollution. Main reasons were identified as urbanization, excessive usage of agrochemicals, pesticides, etc. OECD guideline facilitates testing of chemicals in a mixture of compounds such as contaminated water, but the composition of water should be characterized as far as possible. As such, identification of chemical constitutions in selected waters are necessary. Quantitative occurrence of different chemicals and their substance-specific properties should be indicated. Before the use of the test guidelines for regulatory testing of a mixture, it should be considered whether it will provide acceptable results for the intended regulatory purpose. Ethical clearance was obtained from the Medical Research Institute for this study. General objective: to ascertain the correlation of the water quality parameters with the cumulative mortality rate in 96 hours post-fertilization of the zebrafish embryo. Our specific objectives are to analyze the water composition of Colombo and North Central and compare the findings. Second, to analyze the cumulative embryo death of the zebrafish subjected to Colombo and North Central province water and compare the findings. Third one, to ascertain the correlation of the water quality of the Colombo and North Central province with the cumulative mortality rate in 96 hours post-fertilization of the zebrafish embryos subjected to the Colombo and North Central province water. Under methodology, 30 DUT well water samples were randomly collected from 13 divisional secretariats from Colombo, and 24 water samples were collected from 9 divisional secretariats from the North Central province. Water samples were analyzed for water turbidity, hardness, biological oxygen demand, pH, phosphate, ammonia, nitrate, and dissolved oxygen. These are the wells which were used to collect the water samples from North Central province. All the toxicological assessments were performed using OECD guideline 236. Test guidelines were intended to determine the acute or lethal toxicity of chemicals on embryonic stages of fish. Newly fertilized zebrafish eggs are exposed to the test water for a period of 24 hours to 96 hours. 20 embryos, like 1 embryo per well, were exposed to the test water collected from each DS division. Same procedure was carried out in 3 replicates using 24 well plates whilst exposing 60 embryos to test water. Tank distilled water was employed as internal plate control. Tank water was used as negative control. It was used in 1 plate. 3,4-dichloroaniline in 4 milligram per liter was used as positive control. It was used in 1 plate. Apical points of embryos such as coagulation of embryos, lack of somatic formation, non-detachment of the tail, and lack of heartbeat were observed for 24 to 96 hours. Sublethal and teratogenic endpoints also were recorded in North Central province as well as Colombo water. This first picture was shown coagulation of fertilized eggs, lack of somatic formation in masses of mesoderm is shown at second one. Lack of detachment of the tail, but from the yacht set, it's shown at third picture. This is the lack of the heartbeat. All the test procedure were performed at 26 plus or minus degrees Celsius temperature, pH 7.07 plus or minus 0.12 and 4.6 milligram per liter dissolved oxygen. Eggs were obtained from a colony with a more than 70% hatching rate. Every 24 hours, 4 apical observations were recorded as indicator of lethality. These are the method apical observation of acute toxicity in zebrafish embryos after 24 to 96 hours post-fertilization. Except heartbeat, other correlated embryos, lack of somatic formation, non-detachment of the tail were observed from 24 hours. After 48 hours only, we can observe the heartbeat. The following were considered for validating the test result. Overall fertilization rate of the eggs collected was more than 70%. The water temperature was maintained at 26 plus or minus 1 degree Celsius. Overall survival of embryos in the native control was more than 90% until the end of the 96 hours of exposure. Exposure of the positive control resulted in minimum mortality rate of 30% at the end of the 96 hours exposure. Hatching rate of the negative control was more than 80% at the end of the 96 hours of exposure. The absence of any condition mentioned it, we have to repeat the test. American Public Health Association prescribed standard methods for water analysis, which has used for our research. These are the results of turbidity, hardness, biological oxygen demand, pH in Colombo and North Central Province water, phosphate, ammonia, nitrate, dissolved oxygen in Colombo and North Central Province. These are cumulative embryo death percentage shown in Colombo and North Central Province. Results were analyzed using 2 sample t-test. A significant difference observed in water hardness, nitrate, and phosphate levels in North Central Province compared to Colombo. No significant difference was observed in 96 hours cumulative embryo death percentage. Though sublethal changes were observed, no significant differences were observed in test and control groups. Discussion. WHO recommended level in drinking water. Hardness should be less than 60 milligram per liter in drinking water. Phosphate level should be less than 5 milligram per liter. Nitrate level should be less than 50 milligram per liter. The water hardness, phosphate, and nitrate levels were below the WHO recommended concentration in North Central Province water. Finally, we are coming to the conclusion. Though the water hardness, phosphate, and nitrate levels were significantly high in North Central Province water compared to water collected from Colombo, it was unable to mount a significant impact on zebrafish embryo. Authors thanks ethics and research committees of the Emma Medical Research Institute, and the financial grant was given to carry out the experiments. Staff of the Animal Center is acknowledged for the support extended to carry out this research. Finally, I would like to thank to Salala to give the opportunity to present this study. Thank you very much for listening my presentation. Thank you.
Thank you very much, Dr. Karnakaran, for your interesting presentation. Now the paper is open for discussion. Any questions from the audience? Can I ask 2 questions? First one, now it is a well-accepted thing that it is not necessary to obtain ethical clearance for acute embryo toxicity test if you conduct that is up to 96 or 120 hours post-fertilization. So is there a specific reason for obtaining ethical clearance in this study?
There are so many other administrative issues. So the project may be related to zebrafish, but when we are going to collect water, the project will be analyzed as overall this thing. Without having the ethical clearance, we can't go to the North Central Province and collect the water because this is a very hot topic and all that. So we have to step into the area with caution. So because of that,
and other thing is we got the financial grant from the MRI. So they should know what we are doing really. Because of that, entire project was subjected to ethical clearance, not only considering the zebrafish, the total project was evaluated by the ethics committee as well as research committee.
Right, okay, thank you. The other aspect, now you have checked the North Central Province water collected and also from the
Colombo
Colombo.
district, yeah.
Yes. Now, is there a time difference of the collection of North Central Province water and the Colombo district water? What were the differences in days, for example?
I think only within 1 month, no? Just to
concentrated on the dry period.
I asked from the.
Usually we are collecting the water from North Central Province drying period. Because of that, if raining season, the water will contaminated with other things and diluted. Water will be diluted. So to prevent that, we are collected water from dry period. At same times, we are collecting the water from Colombo district.
All right, same time period.
Yeah, time
And
period.
also the conditions that you store the samples, water samples, it's the same.
Yeah, we are stored the water sample in cool area, then in a refrigerator below the 4 degrees Celsius.
Right, okay. The other question, now you mentioned in the North Central Province water, the nitrate levels are higher compared to the
Colombo water, but even WHO guideline, they are recommended the water level is higher than that North Central Province water nitrate concentration.
Right. No, before incubating these zebrafish embryos in those water samples, have you checked the differences in nitrate levels in these two water samples?
Yeah, there's a separately we are doing to test the water samples to the NARA. We are do the sample for NARA. Then they are from there they test the sample and give the result.
Right, thank you.
I have one question. Can I ask one question? Yeah. Yes. I'm Professor Charita here. Now you mentioned there was no significant difference in the mortality rate between the two water samples, but you also mentioned about like sublethal changes in the embryos. Can you explain what they are?
Yeah, there's a sublethal changes and heterodigital endpoints, which are that one hemorrhages and pericardial edema of that. Other than that's tail deformity. That some features are we are observed during that observation period. But as well as North Central Province water as well as Colombo water also.
Okay, so there was no difference between the two water samples, really, right? Yeah. Okay.
Can I ask another question?
Yeah.
Now, according to the objectives of your study, I think that the interpreting your results in relation to the CKD was one of the objectives. Yeah. So my concern is whether the CKDs develop over a period of time. So do you think this short period you subjected the zebrafish for the research would have given a clear-cut result to come to your objective?
No, in our main objective is our main objective is different from the CKDU to find the CKDU disease. Here, that's our general objective is to ascertain the correlation of the water quality parameters with the cumulative mortality rate in 96 hours post-fertilization of the zebrafish embryo. This is the general objectives. Our specific objectives is to analyze the water composition of Colombo and North Central Province and compare the findings to analyze the cumulative embryo death of the zebrafish subjected to Colombo and North Central Province water and compare the findings.
Okay,
Third
right.
one, yeah, that's a, did you got the answer?
Right. Thank you, Dr. Karunakaran.
Yes, doctor, just one simple question, not question actually, but you mentioned you compared the water quality parameters.
Yeah.
Actually, bacterial or pathogen contamination is also among the water quality parameters. This is just a suggestion maybe, because I think Colombo water is more contaminated and polluted. So if you proceed or continue with further studies, I suggest please check the bacterial contamination as well.
We will consider in future. sir.
Thank you very
Thank you,
much.
Dr. Prasad.
So in the absence of any more questions, we would like to thank Dr. Karunakaran for her interesting presentation. Thank you. Dr. Prasadi De Silva of Uva University, Uva Vellasa University will introduce the second paper and the authors.
The second paper of this session is on cysteine inhibitory capacity of Gunnaroo green seeds, a local variety of Phaseolus vulgaris. The authors are Anushangi DGYR, Vijay Sundara LJMCS, Ampe Mohoti AALT, and Kumari KDKP. The presenter today is Miss Ampe Mohoti AALT. Over to you, Miss.
Greeting dear all. So today I am going to present this presentation. It's about cysteine inhibitory capacity of Gunnaroo green seed, a local variety of Phaseolus vulgaris, under the abstract ID 9 for the 9th conference of Sri Lankan Association for Laboratory Animal Science. So these are the names of my research team, and I am the presenting author AALT Ampe Mohoti, studying at Faculty of Graduate Studies, General Sir John Kotalawala Defence University, Sri Lanka. And first of all, I need you to know about the introduction about this research. So what are proteases? Proteases are enzymes which catalyze the process of proteolysis. Proteolysis is a biological mechanism of breaking down the proteins into simple amino acids. There are several groups of proteases like cysteine, aspartic, serine, metalloproteases likewise, but my major concern is for cysteine proteases. So cysteine proteases are present in everywhere, like animal, plant, and also microorganisms. But cysteine proteases of some infectious hazardous microorganisms playing a vital role in hemoglobin hydrolysis and also blood cell inversion. In addition to that, some cysteine proteases can be involved for proliferation process of some hazardous infectious viruses like HIV. Therefore, those cysteine proteases of those infectious hazardous viruses are known as virulence factors. To overcome all those things, it is beneficial to use protease inhibitors. Protease inhibitors are the compounds that block the activity of proteases. And also, these proteases have the ability to prevent some numerous diseases like metastasis and also tumor progression. Currently, already, we are using synthetic protease inhibitors as drugs and also therapeutic agents for such kind of diseases. But the matter is, we all know that there are some drawbacks of some synthetic drugs as well as synthetic things like long-term and also short-term. In order to overcome those things, our major concern was to identify a natural protease inhibitors, especially from food crops. Therefore, we selected this green bean variety, which belongs to the family Fabaceae. to identify and also evaluate its protease inhibitor content. So this is the objective again in our research. And also, let's step on to the methodology. In here, I have divided the whole methodology into simple 3 parts. In the first part, when sample preparation, after collecting the green beans from Gannorua, beans were sorted, cleaned, powdered into fine particles and prepared 20% solutions by adding distilled water and filtered it well to remove insoluble part and centrifuge in order to get the clear supernatant. After collecting the clear supernatant, we determine the activity of papain inhibitory activity of that plant extract. To do that, actually we used papain and also 1% casein as the substrate. And finally, we added 5% of TCA to terminate the reactions, trichloroacetic acid for terminate the reaction, and then centrifuged it at 10,000 RPM for 10 minutes, and absorbance was measured under the 280 nanometers in the UV visible spectrophotometer. And finally, percentage of papain inhibitory activity was measured by using this mentioned equation. And this is the characterization part. So in here also, I have divided the full characterization part into simple 4 parts. The optimization part, we did the optimization part because we wanted to know what are the ideal conditions to establish the papain assay. That's why. And we selected only 2 factors. One is volume of papain, and also the second one is incubation time. And after that, actually, we were assessed the effect of temperature, pH, as well as the effect of metal ions. Here I have mentioned all the details in detail, and all those 3 things we were selected 5 conditions for each. And in the results and discussion, after the optimization, we identified that maximum papain activity can be obtained from the volume of 250 microliters, and also the 60 minutes as the incubation time period. Initially, actually, we thought that the maximum activity can be obtained from the 300 microliters, but results is clearly showing us the maximum activity can be obtained from 250 microliters. And when considering about the determination of the papain inhibitory activity of the seed sample, there actually we prepared a concentration series from 20 to 1.25. In there actually 10% concentration gave us the maximum inhibitory activity. In there also we thought that 20% would be given the maximum inhibitory activity, but that assumption also was wrong. That the reason for that might be 20% concentration might be contain some other substances like crude protein. That might be the reason. And the effect when considering about the effect of temperature, 37 Celsius was the ideal for inhibitory activities. Since we are dealing with proteins, some extreme temperature like very low and very high might be a cause for protein denaturation. That might be the reason for that. And also when considering about the pH, that also revealed that the 7.4 pH was the ideal for keeping the inhibitory properties of this plant extract. And after adding metal ions, we used 5 metal ions, and it was indicated that after adding barium chloride, we had the highest activity. And also after adding the ferric chloride, we got the lowest activity. And these are the conclusions I summarized. And 10% concentration gave us the maximum inhibitory activity at the 37 Celsius with the 7.4 pH, and there were clear effect on adding metal ions for that. And these are the references I got information for my presentation. And thank you so much for your undivided attention.
Thank you very much. Now the presentation is open for discussion.
Why did you select papain? It is a protease which generally hydrolyze all peptide bonds, no? So your focus is cysteine proteases.
Yes, sir. Majorly we selected papain as the since its availability.
So because usually it hydrolyzes protein into amino acids in a generally. So you are focusing on protease, cysteine proteases. So in your topic also cysteine inhibitory activity, is it right? The topic?
Yeah.
Cysteine inhibitory activity or cysteine protease inhibitory activity.
Cysteine protease inhibitory activity.
Are there any questions from the audience and the other evaluators online?
This is Professor Nayana. Actually, I also had the same question as Dr. Anura mentioned. So now you are talking about cysteine proteases and you are using papain as the substrate. So I was wondering the applicability of this one. Was it done on some literature review or?
Yeah, madam, based on some previous researchers as well as literature review, we decided to select it as our enzyme.
Are there specific protease, cysteine proteases that you can find in the nature?
Cathepsin K and cathepsin K. Most of the time in previous, most of researchers, they use this papain as their enzymes.
So it is not a general, it is not a specific protease. No, it is generally, it will hydrolyze the peptide bond and make amino acids from the protein. No, papain is used to hydrolyze protein usually.
Yeah, most of the times in cysteine proteases, they are using papain. Like for aspartic proteases, they are using pepsin, and likewise there are different proteases for different groups. Actually, we are selecting those enzymes. The major thing we are considering about its availability. Yeah.
Any other questions? No? Okay, thank you very much. So we can move to the next presentation. I'd like to invite Professor Mangala.
Third presentation titled Preliminary Study on Chronic Anti-Inflammatory Activity of Null Herbal Ointment, Jivada, on Adjuvant-Induced Vistarax Hypoedema. Authors are Veerasurya, WHN, Hevavasam, HWDL, Chandrasena, USD, Ratnayaka, WMKM, Kumari, KBKP, Chandrasiri, N, Suresh, TS. Veerasurya, WHN, is going to present the paper.
Good afternoon, everyone, and I'm presenting preliminary study on chronic anti-inflammatory activity of novel herbal ointment, Jivada, on adjuvant-induced Vistarax hypoedema. Mainly, we focused on chronic inflammatory diseases such as arthritis, which remains as one of the world's major health problems. And there is a high prevalence of arthritis and other chronic inflammatory conditions in aged population. And this is considered as a leading cause of disability, adversely affecting their quality of life. And also commonly these chronic inflammatory conditions are treated with non-steroidal anti-inflammatory drugs, analgesics, countervirals, disease-modifying anti-rheumatic drugs, and biologics. But these drugs have to be used for long terms, and that can lead to gastrointestinal toxicity, cardiac toxicity, hepatotoxicity, edema, and nausea. Considering there is no effective remedy for a cure of arthritis like chronic inflammatory diseases, attraction has been drawn towards investigations of the efficacy of plant-based drugs used in traditional medicine in order to save use for long terms. And we can see advantages of herbal medicine over these allopathic medicine, such as lower level of side effects, lower drug interactions, effectiveness in chronic conditions, low cost and availability. And introducing Jeevada is a novel herbal ointment prepared by Fairway Biotech Private Limited using ingredients that have already provided evidence for anti-inflammatory action. This novel ointment contains extract of Areca catechu, Alpinia galanga rhizome, capsaicin, menthol, and camphor as active ingredients. And the Therapeutic Good Administration of Australian government has approved the key ingredients used in this ointment to use as topicals separately. And in our study, the main objective was to evaluate the prophylactic chronic inflammatory action of Jeevada novel herbal ointment on adjuvant-induced arthritis Vistarad model. Our study design was an analytical interventional study, and we conducted our research at Research Laboratory of Department of Biochemistry and the Animal House of Faculty of Medical Sciences, University of Sri Jayawardenepura, and we selected Vistarats purchased from Medical Research Institute, Boralla, and ethical clearance was obtained from Ethic Review Committee of University of Sri Jayawardenepura. And we selected adult male Vistarats, and they were randomly grouped 6 by each, and they were induced with arthritis by a single intradermal injection of 0.1 milliliter of Franz complete adjuvant suspension into the foot pad of the left hind paw. When adjuvant arthritis model, we can use this model as a prophylactic or anti-arthritic model, depending on the starting of the treatment. So in this study, we used a prophylactic model by beginning the treatment from onset of the injection. And the Jeevada ointment and ointment based without the active ingredients were applied respectively to the test group and control group twice a day for 19 consecutive days, and their body weight, thickness of the hind paw pad, and thickness of the ankle joint was measured. Then the results were analyzed by using student test in SPSS software. Measurement of tibiotarsal joint thickness and percentage inhibition of paw pad edema was calculated using these formulas. Let's move on to our results section, and we have demonstrated edema formation in tibiotarsal joint. Here, as we seen here, there was a significant reduction of edema formation in the day 19 in the test group compared to the control group. And we can see the percentage inhibition of edema was increasing from the day 3 to day 19. And there is a significant reduction, and percentage inhibition of tarsal joint edema was gradually elevated, and the maximum inhibition was 77.81 at day 19. And when consider about the edema formation in paw pad, there was a significant reduction of edema formation in day 10, day 14, and day 19 in test group compared to the control group. And weight changes was also monitored as an apparent indicator of arthritis symptoms. There was a significant reduction of weight loss in test group on day 3, day 10, day 14, and day 19 compared to control group in prophylactic study. Discussion and conclusion. The main aim of the prophylactic study was to assess whether there is an effect of new ointment towards the early stages of the arthritic conditions. When considering about the results of our study, new herbal ointment Jivada showed significant reduction of joint edema on day 19 and significant reduction of paw pad thickness from day 10 onwards. And these observations provide scientific evidence for the chronic anti-inflammatory properties of the novel herbal ointment Jivada from early stages of inflammation. And we encouraged commercializing of this ointment followed by clinical studies in order to use this as a treatment for arthritic conditions. And we also suggest further studies to uncover some of the possible mechanisms of action of the ingredients of the ointment. And these were the references I used in our study. And I would like to thank these people. And thank you.
Thank you, Ms. Veerasurya, for your nice presentation. Now the paper is open for discussion. Any questions from evaluators on site?
I have a question. Can I ask a question? Yes. I was wondering whether you used like a positive control in your study, like a.
We conducted this as a preliminary study, and since this is an ointment and there is no actually standard ointment used in arthritic conditions in Sri Lanka, we used only the control and the test groups.
Okay.
Have any idea about active ingredients present in the areca nut? Do you have any idea about the active ingredient present in the areca nut that will ameliorate or reduce the inflammatory responses?
Yeah, the literature shows there is a inhibition of vasodilation, and also it contains some leukocyte infiltration, inhibition of leukocyte inhibitory, and also in other ingredients they have showed some cytokine inhibitory activity, helping the anti-inflammatory activity.
You may have heard about the fact that some scientists have reported that consumption of areca nut may induce oral cancer. So your results are, like a little bit contradictory to the.
Yeah, but mostly that is when we use this orally, but in ointment we don't use that much of concentrations, and the dermal application may not give such kind of result.
So, can I have the next question?
Yes, madam, you can have it.
So maybe I missed that point in your presentation. So what was the route of administration of your Jivada oil into the experimental group?
We applied it dermally. That means this is ointment. So we directly applied it to the foot pad of the rat.
So my question is now, you have used the foot pad injection model, and in the real situation, we will be applying it on the skin, right? As a
Yes.
topical preparation.
Yes.
So do you expect the results to have some differences in absorption?
Yes, ma'am. And we also encourage, we are hoping to do another studies on that also to evaluate the systemic absorption of the drug. And we are hoping to conduct that studies also.
Right. My other question is now, when you use the foot pad injection model, did they by any chance develop any inflammation just after injecting in the foot pads?
Yes, ma'am. We can see that redness and edema formation, and the literature also shows that inflammation is inflammation caused by the injection can be occurred after between 5th and 7th day of the injection. And they also also we can check the behavioral changes also, and mostly the edema formation and the redness of the injection site.
Okay. Right. So how long it took for them to really reduce the inflammation in the foot pad?
Yes.
How long did they take to reduce the inflammation due to the injection itself?
Normally the inflammation come to a peak on day 7, and it exists more than about 30 days, so 20 to 30 days. So we conduct this study up to 19 days, and there was no complete resolution of the inflammation up to 19 days. And we have conducted another study on anti-arthritic effect after 19 days. So it has also shown a reduction of inflammation in the rats.
Right. Okay. Right. Thank you.
May I, actually my question is also about the observation period. We have just observed the animal for only 19 days.
19 days.
So is that done it purposely, or?
Yes, madam. Since this is the prophylactic part of the chronic study, we as per the previous studies and the literature, they have mentioned that up to 19 days we can use for the prophylactic study, and from the day 9 of the injection we can start the treatment for the anti-arthritic study. For that we can observe for more long time, and that study was also done with this study.
Thank you.
I have few more questions. One thing is you indicated that you have included 6 animals per group.
Yes.
How did you select that number?
Since this is a preliminary study, according to the previous studies and the literature, we have selected that they have said the minimum number is 6 per group.
Actually, now it is no more accepted. Right? So there are specific equations for analysis could be done. So you have to go like that. Otherwise, when you go for a publication, it will be difficult. Right? That is one thing. And is there a sex difference?
Yes, we used male rats.
Male rats, only male rats.
Yes.
Right. And the other thing, it is related to analysis. So you have used student t-test.
Yes.
But your timed observations are on different days.
Yes,
That
ma'am.
means day 0, day 7, like that
Yeah.
until day 19.
Yes, ma'am.
For that type of study, can you use student t-test?
We compared it with control test, and in day 3 we compared it control and the test group. Likewise,
it is not advisable to use. Yes, right. So it is the best method is to use ANOVA, and if there is any significance in the ANOVA analysis, then you have to go for another study, another analytical method. Right. Okay. Thank you very much for your presentation.
Okay, the last presentation is on assessment of nutritional properties of a locally manufactured dry dog food and its comparison to an imported brand. The authors are Vimalavira UASL and Pratapasinghe GA. The presenter is Mr. Vimalavira UASL. Over to you, Mr. Vimalavira.
Good afternoon, ladies and gentlemen. Today I'm going to present my undergraduate study. This study was done in order to assess the nutritional properties of a locally manufactured dry dog food and to compare it with the imported dry puppy food brand, which is well established in Sri Lankan market. There's a reason why I did this study, because dry dog food industry is a trending industry globally, showing a compound annual growth rate of 4.1% since 2016. Similarly, in Sri Lanka also, this industry had shown 39.5% growth between 2016 to 2019. Yet, unfortunately, as a nation, we have been importing this dry dog food at a rate of more than 95% of the country's requirement. On the other hand, the literature regarding this type of locally manufactured dry dog food are very rare to find under Sri Lankan context. So in order to fulfill those requirements, this research was carried out in order to identify the nutritional properties of a locally manufactured dry dog food and to assess its growth performances compared to a imported dry puppy food brand, which is well established in Sri Lanka, and also to assess the palatability levels of these two food types. So in order to fulfill this requirement, a feeding trial was carried out between the puppies who fed with the locally manufactured dry dog food and the puppies who fed with the imported dry dog food in order to measure the comparative growth performances of these two food types. So two groups of healthy male mongrel puppies were used for this feeding trial, and they were fed with these two food types for a 4 weeks of time period. Other than that, the proximate analysis result, which include the crude protein, fat, carbohydrate, moisture, ash, and the crude fiber content of these two food types were also measured. The energy estimation of these two food samples were also measured, and the extrude characteristics like bulk density, instrumental color, and instrumental texture of these two food types were measured. And the hair coat condition of the puppies, which fed with these two food types, were measured by a sensory panel consisting of 30 untrained panelists at the end of the feeding trial. And the palatability levels of these two food types were measured by two-pan test. So according to the Association of American Feed Control Officials Dog Food Nutrient Profile, which is published in 2014, a puppy food should contain protein level of 22.5% on dry matter basis, and energy content of 400 kilocalories per 100 grams of food, and fat content of 8.5% on dry matter basis for their optimum growth. Why I selected these three nutritional components? Because these three nutritional components are the most important nutritional components for optimum growth in puppies and the maintenance of adult dogs. So when it comes to the adult dogs maintenance requirement, these values are only be as protein content of 18% on dry matter basis and energy content of 350 to 400 kilocalories per 100 grams of food, and fat content of 5.5% on dry matter basis. So as you can see in these two pie charts, the proximate analysis test result of these two food types had shown the locally manufactured dry dog food contained 18% of protein content and 6% of fat content, while the imported dry dog food had shown 24% of protein content and 10% of fat content. Of course, these values are on wet basis. So if we convert these values into dry matter basis, the locally manufactured dry dog food had contained 20% of protein content and 7% of fat content, while the imported dry dog food had contained 27% of protein content and 11% of fat content. So if you can remember the data that I have shown you in the previous slide, the locally manufactured dry dog food contain less than the required amount of protein and fat content in the locally manufactured dry dog food, but it remains well within the limits for adult dogs maintenance requirement. On the other hand, the imported dry puppy food had contained ample amount of these protein and fat content for optimum growth in puppies. So when it comes to the energy density of these two food types, it follows the same story as protein and fat content. The imported dry dog food contain energy in ample amount for the optimum growth in puppies, while the locally manufactured dry dog food contain less than the required amount for puppies optimum growth, but remains well within the limits for adult dogs maintenance requirements. So because of these reasons, the growth performances of puppies which fed with the locally manufactured dry dog food showed only 0.43 kilograms of weight increasement during the feeding trial, while the puppies who fed with the imported dry dog food had shown 0.6 kilograms of weight increasement per week. When it comes to texture properties of these two food types, which were analyzed by the texture analyzer, the amount of force required to break the pellets of locally manufactured dry dog food is about 103.55 Newton, while the same parameter for imported dry dog food is only 69.23 Newton. So you can clearly see that there's a significant higher hardness in locally manufactured dry dog food, and it is not recommended for puppies to give this much of harder pellet because their tooth structure is not that much mature, but it may be compatible with adult dogs. So possible reasons for this could be the lower amount of moisture content and higher amount of fiber content in the locally manufactured dry dog food compared to the imported dry puppy feed. When it comes to the palatability analysis result of these two food types, the imported dry dog food had shown significantly higher palatability levels than the locally manufactured dry dog food in two-pan test. The possible reasons for this could be the lower amount of fat content and the lack of meaty color and the meaty odor in the locally manufactured dry dog food compared to the imported one, since these three are the major driven factors for puppy to select a food item for their consumption. And when it comes to the sensory analysis result of these two food types, at the end of the feeding trial had shown a better impact from both these food types on hair coat condition of puppies, and no significant difference among them on parameters like glossiness, softness, and optimum coat fill. So this suggests that both these two food types contain essential fatty acid like omega-3 and omega-6 in ample amounts for their nutrition. So when it comes to the theme of my research, it can be concluded that the imported dry dog food is nutritionally more advanced than the locally manufactured dry dog food due to the reasons that I have mentioned before. That means the lower amount of protein, fat, and energy density in the locally manufactured dry dog food compared to the imported one. But on the other hand, the nutritional composition of the locally manufactured dry dog food is more compatible with adult dogs' nutritional requirements. So this would open up a big market as well. Why I say so? Because the puppy stage is only considered as the first 6 months of a dog's life. So rest of the 10 to 12 years of life, we are considering them as adult dogs and they are fed with adult dogs feed. So having a locally manufactured dry dog food, which is more compatible with adult dogs' nutritional requirements, would open up a big market as well. However, further research are essential to clarify those aspects as well. So these are the major references that I have followed throughout my research project. And I would like to acknowledge these people on behalf of the successful completion of my research project. Thank you.
Thank you, Mr. Vimalaveera, for a very interesting presentation. And now it's open for discussion. And the panel members, if you have any questions, please raise.
Have you done a price comparison between two food varieties?
No, sir, actually not the price comparison because I do not did not have the amount of cost that required for the production of locally manufactured food, since that product was manufactured by my research supervisor, Professor Gamtu Pratapasinghe. So I could not do that.
The other thing is, what is the method they use to analyze energy content of food?
Actually, sir, the bomb calorimeter was not working in my department, so I have to use the conversion method. That means the amount a carbohydrate content 4 kilocalories of energy, and the fat content for 9 kilocalories per kilocalories of energy. That conversion factor I had to use for calculate the energy.
The formula.
Yeah, yes.
Have you considered any confounders between two study groups, like their nutritional status and their growth rate can affect with like other factors such as worm treatment and the supplementation of additional vitamins and calciums? So have you taken these things into consideration, the other confounders?
Actually, madam, the puppies were only 6 to 8 years of age, and they were mongrel puppies, and there was not much difference between those puppies. So there was no incidence of any difference between those two puppy groups. Even the initial weight of those puppy groups were same.
During the study period, did you check whether they are, in addition to the food that you provide, did you check whether they have given any other vitamins by the owners? Because if they are providing multivitamins, supplements and everything, it's going to affect your study outcome. Yeah. Did you check that aspect or not?
Actually, madam, what we did was we have collected those puppies and we have kept those puppies in the university premises until in the confinement, actually, until the study period was done. So they were not given anything other than the feed that we were given.
Thank you.
I want to ask you. So you said that the locally manufactured food is comparably, better for adult dogs as compared with the imported. Why do you say like that?
Actually, sir, I cannot say it exactly because I have not tested that one. What I say was just only referring the literature that I have found. So actually, this food is now commercially available in the market as a adult dog food. But however, further research are required to clarify those aspects. So what I just said was just considering the facts that the previous literature. Actually, at that time, we could not find a pure breed dog puppies. That's why we selected mongrel puppies.
Have you obtained ethical clearance?
Actually, madam, I have applied, but the university ethical clearance did not require the ethical clearance because this was not an invasive study, and the puppies were given for the owners after the study period, and still now they are in a very healthy condition.
Although this is not a research study, you can consider it as a kind of clinical trial. So ethical approval is required for this kind of study.
Yes, Dr. Dinan probably might have an idea on that. Do we need
not there?
Yeah.
So how you have selected mongrel puppies?
Yeah.
So how the methodology, you have to explain it better way, I think.
So what we did was
yeah.
we have found the puppies surrounding the university premises. We have requested those puppies from their owners, and they were fed with those two food types for 4 weeks of time period. And after that, we have hand over the puppies to their owners.
So only you have changed the diet that you are
Yeah,
doing.
only the diet, sir.
In that case, yeah, they are not applying the ethical clearance in the nutritional trials. Only for the nutritional trials.
for your nice presentation.
Thank you very much.
Thank you very much for all the presenters who did submit and present their work in this forum. And this is the end of free paper session 3. And we had 4 presentations that is starting from the use of zebrafish embryo model for acute toxicity test. And the one about the P. vulgaris activity that is using the enzyme activity. And also there was one on Vistarate use for the anti-inflammatory effect of herbal ointment. And the last one, it is about the food supplement, or sorry, the comparison of the energy and other components in 2 different kind of food, dog food. So it's a diverse topics that we had during this presentation. And once again, I would like to thank all the presenters for their wonderful job. Thank you.
Thank you all. Now it's time to deliver the token of appreciation. So I cordially invite Dr. Krishanti Premaratna to deliver it to Professor Mangala Gunatilaka. And I now invited Dr. Prasadi De Silva to receive the token of appreciation. Next, we'll move to the very important event of this occasion. So this is time for the certificate awarding. So first of all, I should give my thank to our panel members, our judge panel members, Dr. Anura Jayasurya, Professor Nayana Vijayawardena, and Professor Charita L. Gunasekara. They were waiting, and our Anura sir already present all the 3 sessions, and the other 2 professors participated the whole 3 sessions in online. So I cordially invite Professor Mangala Gunatilaka to give the certificate award to the Dr. Anura Jayasurya. Now it's time to deliver the certificate for our presenters. Now I cordially invite Dr. Krishanti Premaratna to deliver the certificates for our participant presenters. Dr. M.G.T.M. Prasadini. Now I invited Dr. Mayuri Thammitiagoda to deliver the certificates to our other presenters, Dr. K.K. Vijay Sundara and Dr. K.S.U. Kuruppu Arachi. Dr. H. Ratna Divakara. And Mrs. S. Manopriya. Thank you, ma'am. Now I cordially invite Dr. Yureshani to deliver the certificate for our rest of the presenters. Mr. D.P.R.A. Pereira, Ms. C.S. Vikramage, Dr. R. Karunakaran, Ms. A.A.L.T. Ampe Mahoti, thank you, ma'am. Now I cordially invite Dr. Kalpani to give the certificate for the rest of the presenters. Ms. W.H.N. Veerasurya, Mr. UASL Vimalavira, thank you, ma'am. Now it's time to deliver the certificates to our organizing committee. So for that, I cordially invite Dr. Sachini to deliver the certificates. To receive the certificate, first I cordially invite Dr. Gihani Chayavira, the editor of the SLALAS committee. Then Professor Mangala Gunatilaka, Then Professor Sugandhika Suresh, Dr. Mayuri Thammitiyagoda, Dr. Prasaditi Silva, Dr. Yureshani Karunaratna, Thank you, ma'am. Now I cordially invite Dr. Hasanti to deliver the certificate for the rest of the organizing committee. To receive the certificates, I cordially invite Dr. Ramani Karunakaran. And Ms. Risfa Samanuddin, Assistant Treasurer of the SLALAS Committee. And Dr. Kalpani Ratnayaka. And Ms. Chedlia Shah. And Dr. Chandrindu Abekon. Now it's time to deliver the certificate for the assisted staff. For that, I cordially invite Dr. Krishanti Premaratna to deliver the certificates. Ms. Artika Kalaichalavan, WWMPW RB Veerasekara, PADS Palihaderu, then to deliver the certificate for the comparing, I cordially invite Dr. Krishanti Premaratna to deliver the certificates.
Dr. Krishanti is presenting certificate for our comparer, Ms. Shashika.
Now we will hold the session to get to know the winner of the competition today, the presenter who is the best presenter. Until we get the result, we will hold the session for a little while.
for Laboratory Animal Science. The award for Best Oral Presentation for Dr. Shamini Jayasekara Award of 9th Annual Scientific Sessions and International Conference of Sri Lanka Association for Laboratory Animal Science goes to pre-paper number 2, Preliminary study on alteration of hepatic macrophages by crude leaf suspension of Obesaki octanda, Heen bovitia in thiosate-induced rat liver cirrhosis, authored by Bogahavatta BRS, Kodituakku SP, Vijay Gunawardena MBP, and Vijay Sundara KK. I cordially invite our panel of judges. On behalf of the panel of judges, Dr. Anura Jayasuriya on the stage to award the certificates to the winners. And I cordially invite our president, Dr. Krishanti Premaratne, to accompany our guest to the stage. Since we don't have the presenters on the venue today, I cordially invite one of the members from Faculty of Veterinary Medicine, University of Peradeniya, to collect the certificates on behalf of the authors. Ladies and gentlemen, round of applause for the best oral presentation for Dr. Shamini Jayasekara Award of 9th Annual Scientific Sessions and International Conference of Sri Lanka Association for Laboratory Animal Science. Thank you very much, sir. With that note, we conclude the 9th Annual Scientific Session and International Conference of Sri Lanka Association for Laboratory Animal Science. Thank you very much.
Thank you, Gihani. After 2 long days, we have come to the end of the 9th Annual Scientific Sessions and the International Conference of SLALAS. First of all, I would like to acknowledge all the members, participants who are here today during these 2 days with us. And this type of an event will not be possible without the financial assistance. Therefore, first of all, I would like to show my gratitude towards the ICLAS for providing all the financial assistance. For that, I would like to thank Prof. Cynthia Picau, Dr. Montip, and Prof. Mangala Gunatilaka for assisting to obtaining this grant and conduct this type of a hybrid event, because with the COVID situation, we were unable to physically meet during the last time. But fortunately, this time we were able to meet and conduct this conference physically and also online. And also, secondly, I would like to thank Dr. Prasad De Silva and all the resource persons who contributed for the pre-conference workshop on advanced zebrafish model. That was the pre-conference workshop, which was in parallel with our conference. I would like to thank all the people, all the resource persons helped in that regard. Also, I would like to thank the other resource persons who have assisted us throughout this year when we are conducting different types of workshops, the certificate course, the zebrafish course, and cell culture workshop, systematic review workshop, all these para and arrive guideline workshops. Some of the workshops were conducted virtually, some of the workshops were conducted physically. For all the workshops, all the resource persons who contributed, I highly appreciate, and also the EXCO members who have assisted in conducting these workshops, I appreciate it. And I would like to add a special note regarding appreciation for the Medical Research Institute for conducting all the practical sessions, and also the Colombo Medical Faculty for providing us the Zoom assistance and providing assistance for conducting the systematic review workshop. In addition to that, I would like to thank the media team who have helped us conducting these hybrid events successfully. Also, I would like to thank Mr. Lasit. I don't know whether any of you know about him. He's the person who has helped me with designing the conference flyer and all the workshop flyers. And Prakash, who has designed the zebrafish workshop flyer. They have immensely helped to publicize these events very nicely with their design and creativity. Also, the Scientist magazine, is also appreciated, because when we want to get the comments for the guidelines document, consensus guideline document, they step up and they provided assistance to get that contribution. So I highly appreciate the contribution given by the Scientist magazine. Finally, I would like to thank all the judges who have helped us judging the presenters who have presented today, all the presentations. In spite of their busy schedules, they have been able to come here, and two of them have joined online, and they have rendered their duty as the judges, and they have provided that service for us. And all the presenters, I highly appreciate their contribution, and I hope next year's also, when we are conducting the annual sessions, that you will be participating with our annual sessions. With that, I would like to officially conclude the annual scientific sessions of SLALAS. Thank you so much, and joining with us for the tea. And our AGM will begin at 3:45. Thank you so much.